Hello, everybody. Thank you for being here with us in this incredibly interesting symposium. I am Chiara Saracini, academic researcher in the Catholic University of Maule in Talca, Chile, and I am honored to see people from many countries joining this event. This is the one and only positive outcome of this pandemic. It allowed us to explore new ways of virtually gathering and learning despite the distance. This event is organized in the frame of my research project, Fondecyt de Iniciación. So it is founded by the ANID, the Chilean National Agency for Research and Development. I have the honor to present today 6 researchers who will address some of my favorite topics about cognition, such as brain connectivity, oscillations, dynamics, and above all, consciousness. I am sure that you have read some of their works, since they are well known in their area. Before introducing the first speaker, I would like to invite the director of the Research Center for Neuropsychology and Cognitive Neuroscience, CINPSI Neurocog, which I am part of, Dr. Boris Lucero, to say some welcome words.
Thank you, Chiara. Good morning for those in Chile and good afternoon for those in the north of our beautiful planet. Thank you to each and every one of you for being here with us today. We are very pleased to be able to welcome our speakers and guests and all the audience interested in the study of this marvelous field. And today marks one significant event with this fantastic symposium on brain connectivity, neural oscillations, and dynamics of cognition and consciousness. And we are proud to be able to host it today here at our research center, CINPSI Neurocog in Talca, Chile, with all of you. I would like to express my sincere appreciation to all of you who generously helped us make this event come together to become a success, especially Dr. Chiara Saracini and all of our dear speakers. Our research center has been working intensively with electroencephalography, so each of the topics that will be reviewed in this symposium today is of our utmost interest. We believe that this interest is shared with a growing community in the cognitive neuroscience field, so we hope that all of you can take advantage of the knowledge generously shared by our speakers. Well, I don't want to take too much of your time. We need to leave time for the great topics that we are going to hear now. I am sure that today will be fun and full of learning. So thank you very much for listening, and let us welcome all of you to this symposium. Thank you and enjoy.
Thank you, Boris. I must say I'm also very excited about this symposium. Besides, this is the first time that I organize one of my own. We will have 3 presentations first, and then a Q&A session, then a small break, and then other 3 talks and a final Q&A session to let the invited speakers answer your questions. And we hope that you have many questions. So, without losing precious time, I am going to introduce the first speaker. Andres Canales-Johnson is a researcher from the Department of Psychology of the University of Cambridge and is also a colleague of the Catholic University of Maule here in Talca, Chile. He investigates on the brain dynamics of internally generated conscious phenomena, such as perceptual ambiguity, mental imagery, learning during sleep, hypnotic hallucinations, visceral consciousness, and the brain dynamics of vegetative patients during the process of losing and recovering consciousness. Today, he will present his talk titled Dissociating the Contents of Consciousness Using Brain Information Metrics. So I leave the word to Andres now.
So good morning, everyone. It's a pleasure to be here. And today we're going to talk about a bit of the research that I've been doing, I would say, for the last, 10 years or so. And the title of my talk is Dissociating the Contents of Consciousness Using Brain Information Metrics. The main idea of the talk is to try to discuss how we can use connectivity metrics, different connectivity metrics, to understand what I call internally driven perception. And as an example of what is not internally driven perception, here I have an example where, for instance, in a normal setting, the kind of experiment that we do, we present stimuli over and over again. Let's say this grumpy cat versus a house or versus another grumpy cat. And what we usually do is try to understand or to investigate within the brain activity, what are the neural correlates of seeing, let's say, a cat versus a house. I call this kind of approach the perturbation approach, because what you're doing is basically to perturb the brain with different images at different time points. And in this kind of framework, you know exactly where the onset of the stimuli is. So in that sense, it is easier to investigate because you know exactly when things are presenting to you. And then you can track in the brain what is the response to that. A different approach would be to ask yourself, okay, what happens with those kind of mental phenomena that are not externally driven, like presenting you a face on a screen or a sound, but they are more internally driven. And we know for a long, long period, based on different authors, including Varela and Walter Freeman, that perception is not something that is the passive absorption of information from the outside, but rather is some kind of internal, let's say, interaction between what happens in your brain, what happens with the endogenous activity of the brain, and the incoming information from the external world. So how can we study phenomena that are not necessarily externally driven, but are mainly driven by what happens inside the brain? And here I have another example. Let's say that now I ask you not to see the face of the grumpy cat and the house, but to imagine them. Then the question is, where is the onset of this? So when you actually start imagining and when do you actually end up doing so? It's not clear anymore. So the onset of the content that is not now externally driven, but is mainly generated by the subject in the act of imagining is not clear at all. So how do we study this? So I would argue that this is a bit more complicated to study, but we have tools to do that. So in this talk, I'm going to talk in the first part about the study on visual imagery. And in the second part, I'm going to discuss another study on ambiguous perception, which is another way of studying internally generated perception. So in this study from 2021, what we basically did was asking participants to imagine faces that they studied previously, that they were celebrities. And what we basically did was presenting the name of the celebrity, then an oval on the screen, a gray oval, and then we asked them to imagine the image of the face of that celebrity and to press a button when they have the most vivid image in mind. And we had a control condition where they just observed the name of the celebrity and only saw the oval on the screen. So the idea was to contrast two processes, one in which you're only seeing something on the screen versus when you're seeing the oval and projecting the imagined image of this specific face. So you have a contrast between the imagination of the face and, let's say, nothing but a gray oval. And what we're interested in was, okay, to study what happens in the brain in terms of how different networks communicate information in time. And this is, I think, the important bit for this part for the current symposium, is that there is a way of studying brain communication by measuring in the brain activity what we call phase coherence. And the main idea is that if you record electroencephalography and you record the brain activity from these tiny electrodes in the head, what you can do is to separate different waves. So basically, if you actually think that the brain works in different waves at different frequencies, you can separate these different waves from waves that are really, really fast and low in amplitude, like this gamma range that oscillates around 30 to 60 times per second, to very, very slow waves like these theta waves that they are higher in amplitude and lower in frequency at around 4 to 5 cycles per second. So you can separate the brain activity into waves. We were particularly interested in the gamma range because it's mainly related to perception, so to the function of seeing, and also with the theta waves that in this case are more related to the act of remembering the face and try to evoke the face on the gray oval that I just showed you. And what we measured with the EEG was what is called synchronization between brain waves. And the main idea here is that if you have two electrodes, let's say an electrode in this area and in this area, what you try to capture with this metric is how aligned the phases or the timing of these two oscillations are. And we will call this a highly synchronized pair of electrodes because the signals, basically the peaks, coincide between the two, versus this case where the synchronization is really low because the peaks are not aligned between each other. And my metaphor for this is like jumping together. So if you think about synchrony in the brain, it's like if we all would be jumping together with the same pace. So these two areas, by the mere act of jumping together, they are communicating information. So what we did was basically to study what happened in the imagery versus the control 1.5 seconds before people press the button. Remember that the button here represents the moment when they have the most vivid image of the imagined faces. And what we did was basically to record the intensity of this synchronization at these frequencies for the two conditions. So here in A, what you see at 0 is the moment when they press the button. And we are interested in what happened 1.5 seconds before that. The same with the control. And if we care about what happens with the synchronization in the theta range, where these slow oscillations are going on, what we see if we contrast the imagery versus the control is that there is an increase in theta phase coherence in the frontal electrodes that is higher in the imagery condition than in the control. So that effect happens 1 second before they press the button. So before they actually signal the moment when they have the most vivid image in mind, there is an increase in the communication at this frequency range in the brain. What happens with the higher frequencies, with the gamma? Well, we see something similar, but now we see it around 200 to 250 milliseconds before the button press. And again, we observe higher synchronization in the gamma range in the imagery condition versus the control. So we have two effects here. When you're imagining a phase, you see a transient effect in the high frequencies 300 milliseconds before you have the most vivid image in mind. And you also have a long and sustained effect in the theta range, almost 1 second before you press the button. So the conclusion of this part is that the visual imagery of faces is associated with this spatially broad phase synchronization pattern in two frequency ranges, theta and gamma, arguably involving phase memory retrieval in the theta range and visual binding in the gamma range. So this is an example how you can study phenomena that are internally generated, like the imagery of a face, and see what happens with the brain correlates. So now I'm going to talk about ambiguous perception. And this is another example of internally generated phenomena, because if I show you this picture to you, ambiguous perception basically means that whatever information comes from outside can be interpreted in different ways. So for instance, this number can be interpreted as a 6 or as a 9, depending on where am I standing on, or this kind of illusions where I can count 4 or 3, let's say, dogs, or the classic illusion of these vase versus two faces illusion, where you can either perceive one vase here in white or two faces here in black. So the interesting thing about this phenomena is that the stimulation is the same. So you have exactly the same image in front of you, but you can interpret it as two different things. So the interpretation, let's say, comes from within, while the constant image is in front of you. And there are very good reasons to believe that this kind of phenomena is useful for neuroscientists, and one of them is related to study the phenomenology, which is the ability to understand which of these two interpretations are you actually experiencing. And the idea that we're going to use are two conceptual ideas coming from, let's say, the 90s. They are very popular now in a theory called integrated information theory. And the main idea is that you can perceive everything around you as integrated, as a single unitary thing, or you can also make distinctions. Let's say if you have this image, you can experience this image as a unitary percept that we're going to call the integrated percept, that is this vase, or you can also perceive it as two different contents, which are the two faces that we're going to call the differentiated percept. So our perception in general jumps from fully integrated to differentiated. And the idea that we had was, and it's a very, very empirical question, is does perceptual integration, so the ability to integrate, goes hand in hand with brain integration, with some metric in the brain that captures integration? And thus perceptual differentiation, this ability to segregate the stimuli, let's say in these two phases, go hand in hand with a metric of brain differentiation. So can we have a map between your experience, integrated or differentiated, and what happens in the brain? So brain integration and brain differentiation. So we asked this question, and in this paper of last year, what we did was basically having a stimuli that is not visual, but is auditory. But let me explain you how does this work. So what we did was presenting a series of tones that sound like beep, 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 and so on and so forth. And the idea is that if you hear this for a minute or so, you will start experiencing two types of grouping, two types of percepts. You can experience this series of tones as an integrated percept that sounds like a galloping rhythm, tak-a-tak, 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 that is gonna alternate with another percept that is experienced like they call it like a Morse code, because you hear the upper tone in the foreground and the lower tone in the background. So basically, your perception alternates between one single object into two objects, then into one object and two objects. So this is interesting because we can now test our hypothesis that this kind of percept, when it's experienced as one, it should correlate with higher integration in the brain. And this other percept, when you experience it as differentiated or two objects, should go along with higher differentiation in the brain. So what we do is that we ask participants to press a button every time with one hand, every time that they feel that the integrated percept changes into two, and another button when they experience that the two objects changes into one, and so on and so forth. This time we did a bit of phase coherence, but we cared more about metrics coming from information theory. Why? Because I'm going to try to explain why. But for now, what you need to know is that how you can measure now brain integration. You can do it with this metric that is based on mutual information. I don't want to go into the details, but if you allow me to make another analogy, you remember that I said that with the brain waves and with the neural synchronization, you can think of people jumping, like jumping together. That implies that in some way, everyone is doing the same thing at the same time. This other metric is more like dancing together, and it's more like tango. And the idea here is that during tango, the two partners that are dancing, they don't need to be doing exactly the same thing over time. They can have different positions at different time points, but they're still coordinated. So this metric is a way of capturing coordination between different brain signals that is not necessarily what we call linear or proportional. It can be more complicated than that. So we use this metric of, like, say, the dancing approach to study in this current study as a metric of information integration. And we also use this other metric that we need to have for testing brain differentiation that relies on the idea of compression. So if you have an EEG signal like this, the main idea here is that you take your EEG signal, your brain signal, and you try to compress it like a zip file, literally like a zip file, like this kind of zip algorithms that we use for compressing our photos in our computer. We use it in exactly the same manner in the brain. And the logic behind that is that if your signal is very, very, very repetitive, so it's not differentiated enough, then you should be able to compress it really high. On the other hand, if your signal is really, really differentiated, so it has a lot of different information patterns, the compression shouldn't work that much. So the more you compress the signal, the lower the differentiation of the information is. The less that you can compress the signal, the more differentiation you observe in there. So we can use it as a proxy of information differentiation in the brain. So these are the main results. So basically, I'm gonna show you first what happens with the metric of integration. So check the left side of your screen. So the main idea here is that when people experience these sounds as an integrated percept, so as one object, what we observed in the brain, if 0 is the moment when they press the button, and we check 2.5 seconds before, is that this metric of neural integration tends to go up. And it happens between the frontal and the parietal electrodes. So you see an increase in information integration between these two areas when people experience integration. So perceptual integration and neural integration go hand in hand. Now, what happens with the neural differentiation? So when people report now the percept as two objects, so basically instead of having one, you have two, so the perception is more differentiated. Now what you see is the opposite. So you see now that the metric of differentiation in the brain goes up before they press the button. So perceptual differentiation goes hand in hand with an increase in neural differentiation. So the two processes, perceptually and neurally, are matching together, which is very interesting, I think. We also have a control that I'm not going to get into the details, but if you actually manipulate the sound in a way that you make the two percepts changing physically, not in your brain, because remember that this is a bistable perception. If you actually make them change in the stimuli, you don't see any difference in any of the two markers. So these changes are mainly driven by your interpretation of the stimuli and not by changes in the stimuli itself. We repeated in a participant who had electrodes in the cortex, so using electrocorticography, not electrodes in the surface, but electrodes inside the brain, and we observed exactly the same effect. So this can be replicated using electrodes in the frontal and in the parietal areas as well. One thing that I want to mention, and this I think is nice for discussing later on maybe, is that the classic metrics of phase synchrony, so like jumping togetherness, they didn't mark any difference between the two types of percepts, neither in the EEG nor in the patient. So these metrics that were very useful during the imagery study that I show you are not sensitive enough to capture these endogenous changes in the current study. Only the information sharing approach was capable of doing so. Why would you say? Well, I don't have time for this, but Laura Imperatori in 2019 showed that these metrics of information, apparently they capture much better nonlinear interactions in simulated brain activity. So you can take a look at this paper. So the conclusion of this part is that perceptual integration and perceptual differentiation, so basically whether you're actually experiencing the object as one or two objects, can be indexed by an increase in the corresponding neural marker of integration and differentiation in this frontoparietal axis. Now to conclude, let me show you a spin-off of this study, but it's about ambiguous perception, but without pressing buttons, without report. Why is that important? So the thing is that problematically, the study of these internally driven contents require participants to report about their experience, to report what they listen or what they see by usually pressing buttons. And in the last 6 to 7 years, the people have been complaining about the fact that these neural markers can be confounded by the fact that you are actually making a decision about the stimuli. So in other words, these neural markers that I just showed you can be related to the actual perception, but they can be also related to the act of reporting, so post-perceptual processing. If you see something or if you hear something and then I ask you to press a button, there are a lot of processes that are unrelated to perception itself, like making a decision, asking yourself, what am I seeing, and then making the motor response, that are not necessarily related to perception. So can we separate the two processes? Can we separate perceiving from reporting? And if so, how do we do that? So what we did was running an experiment now in a very similar way, where we have now visual stimuli that can be experienced as one object or an integrated object, or as two different objects. I don't have time to show you the stimuli, but basically these are these kind of grids that are moving all the time. And since they're moving, the illusion is that you can perceive them as one thing moving up or as two grids moving apart. So again, you have one integrated percept or two differentiated objects. But the key thing here is that we have a condition when they had to report, again, as I showed you previously with buttons. Well, we have an independent measure that is based on the eye movements. And I think the cool thing in this study is that we managed to use this ocular activity to track which percept the subject was experiencing without asking them to report. So we have another condition where they don't have to press any button. They just observed the stimuli. And then based on the eye movements, we could tell whether they were about to experience the integrated one or the differentiated one without asking them to report. It's like mind reading. So we have a condition when we ask them to report and another where we didn't. And we infer what we're seeing based on the eye movements. And there is a lot of behavioral stuff that I'm not going to show. I'm going to jump immediately into the brain activity, just as an example of connectivity metrics that you can use. And again, we use these information theory metrics. As a measure of information integration, what we used was a metric called transfer entropy or directed information that what it tells us now is not only whether two electrodes are sharing information, but the direction of the flow. So now you can know whether, let's say, frontal electrodes are directing more information to the back, or for instance, you can ask whether parietal or visual electrodes are directing more information to the front. So you have an idea of the directionality of the information flow. And we use our classical now measure of complexity or compression as an index of brain differentiation. And what did we find? So these are the results for the information flow. So what you see here in the upper row is the information going from the front to the back of the brain when people are reporting the integrated percept versus when they are reporting the differentiated percept. And this is the contrast between the two. And what you see is that when people are reporting the integrated one, you see more information at different delays between the front and the back. So you have an overall effect of higher front to back information when people are experiencing one object versus two. The interesting thing is that there is nothing coming from back to front. So when you test what type of, whether there is flow of information between back and the front, you see nothing. And this is expressed in this interaction here, where the integrated percept has higher back to front information than the differentiated one, but not the other way around. So apparently this ability to change between percepts is something that is, I would say, higher in the hierarchy of processing. It's something that is mainly driven as a top-down modulation from front to back. The interesting part is that this, remember, is when they're pressing buttons. What happens when they don't press buttons? It happens something very similar. So if now you infer the percepts based on the eye movements, so without asking the participants to report, you see something very similar, but at different delays, if you want. Again, you see higher information flowing from front to back in the integrated case and the differentiated case, and you basically see nothing coming from back to front. So the kind of interaction that we see is very similar for this neural marker when you compare the report versus the no report case. Now, what happens with the differentiation metric? It happens something very similar than in the auditory case. So when you report two objects instead of one, so when you report the differentiated percept here in blue, you see an increase in differentiation in the brain in the frontal electrodes. that is higher than when you report 1. So again, perceptual differentiation goes along with neural differentiation. And we didn't observe this in the back. However, the interesting thing is that when you do it with the eye movements, the effect disappears. So when you infer the percepts using the eye movements instead of the button presses, this effect is gone. So there are no differences anymore, either in the front or in the back. And this is reflected in the interaction as well. So what does that mean? So I would say that what this means is that our previous marker of perceptual contents that I show you in the auditory domain, it's only there when you have to report. So our conclusion is that the metrics that we use for studying perception are not necessarily all about perception, but they also can be signaling post-perceptual processes that we were not aware before because we didn't have a kind of task that separates the two. So what we want to claim is that this front to back information is actually a marker of perception because it's present in both conditions, when you press buttons and when you don't press buttons. While our marker of perceptual differentiation, since it's only present when you press button and it's absent when you don't, we think that is mostly related to post-perceptual processes. We have no idea which one, but not directly to perception. So as a final conclusion, as you might have seen, I would claim there is not a straightforward relationship between the contents that we observe in the mind and simple neural metrics. However, it is possible to find neural signatures by using connectivity metrics that not only depend on the phase or the classical spectral activity, but also on information and complexity. And these two concepts of integration and differentiation can help us to map or to frame some kind of theoretical background for understanding the perceptual contents. So finally, I want to thank everyone, all the people that have been involved in these projects, in particular the lab in Cambridge, the lab in Amsterdam that I collaborate with, my collaborators in Glasgow, and of course, my collaborators in Chile, Boris and Chiara. If you have questions, I'm usually on Twitter 24/7. That's my Twitter. And it's been an absolute pleasure. Thank you very much.
Thank you, Andres, for the interesting presentation. So I'm going to introduce the next presenter, Srivas Chennu. He's a lecturer at the University of Kent and a senior research associate at the University of Cambridge in the Department of Clinical Neuroscience. As a computer science and engineering researcher, he develops methods for computational connectomics, applying data science and machine learning to build population-level models of brain connectomics, which have applications in neuroscience and medicine. His research brought him to span from India to Germany and UK, and today he will be with us with his talk titled Contrasting Functional versus Effective Connectivity in the Human Brain, Evidence from Physiology and Pathology. Srivas, please take the virtual microphone and the stage is yours.
Excellent. So thank you for that amazingly detailed introduction. I don't think I deserve it, but here we go. So as you've already kind of introduced, a lot of my work is coming to neuroscience from a computational perspective, and that's kind of really where I should start. So about myself, really. I'm currently based across 3 different places in the UK. Cambridge, where of course I have the pleasure of working with Andres and other people, brilliant neuroscientists, and at the University of Kent in Rochester, where I sort of can pretend to instead be a computer scientist, because a lot of my background and the kind of focus I have comes from a computational perspective. And as was also been mentioned, I work in machine learning in the UK in the sort of corporate sector. So I kind of have many hats. Each of these places are beautiful to visit, so please do if you can. Here's a picture of Cambridge, and Andres goes boating on the lake and the river. almost every week, I'm sure. You've got London to visit, and of course, Rochester. So I hope to see you here at some point when we sort of can travel. And today, what I want to do is sort of bring together research across all these 3 different centers that I have been conducting now over the last 10 years or so, primarily to ask some broad questions around what does it mean to study consciousness, and what tools does science have to bring to answer that question. And I'll show you examples of where case studies of these sort of applications have some real value and sort of contribution to add above and beyond sort of the core empirical perspective in neuroscience. So let me start with the problem statement at a very, very high level, and then try and drill it down. Many of us have a shared interest in the question of what are the neural correlates of consciousness? This is one of the biggest questions of our century. You know, it's been sort of named as some of the big challenges in science because we have discovered lots of things about the universe, but we don't know quite what's going on between our 2 ears. So what goes on in the brain to generate consciousness? This is the question. Now, it's an underspecified question. So people boil it down to, okay, what's tractable or meaningful to answer using neuroscience is what are the states of the brain that correspond to states of and contents of consciousness? And Andres has spent a lot of time telling you about this. And further, and this is what I am also interested in on top of this, is that what models, and I mean models in a very technical or computational sense, as I'll show you, what models can we use to describe the state of the brain when consciousness has a certain state or a certain content? So what is modeling? So this is a term that often sort of is loaded and overloaded because different people mean different things. So when I say modeling, I'm thinking of models of these kinds. So a discriminative or a descriptive model is basically where you have some data and you want to infer some hidden truth about what's going on inside the brain based on the data you can observe outside the brain. And you basically go straight from the observed data to that hidden truth. Such a model is a discriminative model, and you can use basically a lot of the stuff Andres talked about in terms of calculating power spectra, information theoretic metrics, roughly fall into this category because you take the data and you infer some parameters from that directly. Even machine learning models of many kinds, like linear regressions and all that stuff you might have heard about, neural networks, they all fall into this category. A generative model, on the other hand, is a model where instead of going straight from the data to some hidden truths, you first kind of posit, you sort of postulate what kind of data you would observe under certain truths if you were to be able to observe them. So that's essentially a P of X given Y. And then you invert that. Now I will show you in my talk what the differences are between these real examples and why one has reasons to prefer one over the other in different cases. So I know this is very, very high level and some technical concepts are being introduced, but hopefully I'll get a chance to tell you more about this with examples. So let's start with the first thing. So let's start with exactly kind of what Andres picked up about neuroscience, which is, okay, let's imagine we collect some data from this lovely participant here. We have this net of EEG electrodes on our head, and we're collecting 256 channels of data, which is a lot of data. Great. When you take that data and you first represent it as a grid of electrodes on the head, and then what we can do is we can take that and represent the information in that signal using things like frequency decomposition, and you might have heard of things like alpha waves and beta waves and so on that EEG is typically decomposed into. And then you take that signal and try and measure something about the relationship between those signals to say, okay, if the relationship between the signals looks like this, it must mean this about the brain or that about the brain. But the signal itself just carries information on, okay, it behaves like this or it behaves like that. In of itself, it is not the brain, it is some sort of indirect cause of what's going on inside, right? So that's kind of essentially what something like a sort of metric like the weighted phase lag index, which is a common and popular metric in this space, is trying to do. It's trying to measure the relationship between the signals. And an alternative way, which is what Andres mentioned to you just now, is to take the same EEG signal, but instead of calculating spectral properties of the signal, we can calculate information theoretic properties and then calculate some sort of information theoretic communication metric about the brain and say, well, I can build a pattern of information sharing in the brain and try to infer or connect that with brain state. The alternative way, this is complementary, it's not either or, but a sort of different way of thinking about the problem is to say, okay, I've got some EEG data, that's on the left side, some observed data. What I'm really interested in is what is the brain doing that generated that data? Now, the way generative modeling thinks about this kind of problem, and a particular example of generative modeling is what's called dynamic causal modeling, which many of you might have heard about, is where first you assume, well, let's imagine that the brain is like this, and you can make reasonable assumptions about the brain based on neurophysiological data, based on the physical architecture of the brain, and say if the brain configuration at any given point is of this kind, and that is specified in great detail in dynamic causal modeling, involving specific neurons and pyramidal cells and interneurons and so on, which I won't go into in detail in this talk. We simply won't have the time. But at a very high level, what it's saying is, imagine that the brain has these populations of neurons, and they're connected in this particular way, and those connections have specific strengths. If that is true, this is the data we would expect to observe. So that's called this forward problem. So predicting the data from some assumed brain state. And then the inverse problem is, given a brain state, trying to infer the parameters of that hidden model. For example, what is the connectivity between certain populations? And what generative modeling does is basically does this loop of inference going from the data, so going from the brain state to the data and back. So it's a sort of a really distinct way of modeling the same data. And I'll show you what value it can bring. In particular, you might ask, well, what can I do about this with this sort of modeling technique that is different from what I can do with the kind of connectivity estimation methods I showed you earlier? What you can do is make stronger and more directly causal claims. So for example, the well-known mismatched negativity, which is sort of a very well-studied event-related potential in the EEG signal observed outside the brain, DCM can be used to try and model that signal to show that very specific networks in the brain are involved in generating that signal. Now, it's not possible to do this just by doing connectivity analysis using either spectral or information theoretic measures. They can only tell you how the signals are sort of going with each other, but they can't tell you what in the brain might be causing that. So this well-known paper and many other papers since that we've also worked on show that you can basically use things like DCM to infer the structure of the brain networks that are generating externally observable data. So summary, descriptive versus generative modeling have complementary value. One is measuring functional connectivity, what goes with what, right? So you can answer questions like what pattern of connectivity goes with or correlates with this state of consciousness, whereas effective connectivity is trying to answer what pattern of connectivity inside the brain is causing this or sort of state of consciousness, trying to make some causal claims, which is what kind of lends it its power. But it doesn't mean it's always the better thing to do. Generative modeling has challenges because of the fact that it's doing sort of a greater depth of inference. It's also computationally more complex, and it's harder for many cases to know what kind of models in the brain that one needs to formulate to be able to explain the data. Because unlike in the descriptive modeling situation, the generative modeling approach only works if you can first formulate a model that you think is a good model of what's going on inside the brain. If we have no idea what might be going on, it's kind of difficult to start doing generative modeling. So typically, people start with descriptive modeling, use that as a means to come up with sensible generative modeling. Okay, so let's keep moving on. What I'm now going to do is give you some examples of where both of these techniques in my own research, I found very useful. Hopefully, that will connect with some of the challenges that you're also facing in your own research and your own work. So one sort of situation is trying to understand what the brains of patients who have had brain injury might look like. So to give you a sort of landscape perspective on this whole challenge, right now, certainly in Chile, because it's earlier than in the UK, you're all fully conscious, fully awake. After lunch, like me, you might start slowly kind of drifting off to sleep, especially if the talk is boring, right? And then over time, in your life, more generally, you might have some opportunity to experience what general anesthesia is like, where you're getting an anesthetic to make you completely unconscious, right? And hopefully this happens to nobody, but there are patients who are in this really, very weird situation where they have a certain level of wakefulness. They're not in a coma, but because of the injury to the brain, they appear to have no content of consciousness. They're awake and their eyes are open, but there doesn't seem to be anybody there. And this is called this unique disjunction called the vegetative state, which has been very challenging to understand from a neuroscientific perspective. But it's not just from a neuroscientific perspective that we need to understand it. What's been shown is that after injury, some patients make a really good recovery, like the patient here on the top, for whom I'm showing structural connectivity scans using what's called the DTI, tensor imaging, where basically patients, in some cases, suffer injury. As you can see, their connectivity is reduced compared to the control on the left. But over time, by 1 year after the injury, they have recovered a lot of that connectivity. But some other patients, they just go downward. They degrade and eventually die. And we don't quite know why. So if we can figure out how therapy and treatments and interventions can help, we can perhaps enable positive outcomes. So neuroscience has been interested in this problem for that reason. We too have looked at this using the same techniques I talked about, EEG, where we put these electrode nets in the patient's heads and measure the spectral patterns of connectivity in their brains, and then essentially convert that to a connectivity map, kind of like what Andres talked about as well. Take those connectivity maps, and here I can see I'm sort of zooming through a lot of the methodology in the interest of time, but essentially what you're taking from the connectivity map is some sort of representation of the connection of the brain, which you can plot in this beautiful kind of picture like this, which is essentially the kind of functional connectivity map of what goes with what in the activity we can observe in the patient's brain, and just coloring and sort of drawing lines between which electrodes seem to be connected to which other electrodes. It's saying nothing about what's going inside the brain. We don't know that, but we can observe something outside and make pretty pictures like this. But not only can we do that, we can then do some quantitative analysis using mathematics to try and capture the nature of the network in the brain, nature of the network that's observed in the EEG using things like graph theory, which is what we have done. And if you do this, and you can basically, just as a case study, show that when somebody gets an anesthetic to basically lose consciousness and then recover, for example, during surgery, this pattern of connectivity you can observe outside the brain, where it has a very systematic pattern. First of all, when you're awake, you can see this kind of mohawk kind of pattern, which basically has a frontoparietal axis, where there are strong connections across the front and the back of the brain, which dies away when you're sedated. And then when you recover from sedation, it comes back. Now, I've said nothing about what's going on inside the brain that generates this pattern. All I'm saying is, oh, look, when the person is unconscious, it looks like this. When they're conscious, it looks like this. There are no claims being made here about what's going on inside, because we can't know directly from the pattern of connectivity. We can only say something about the regions. So in the same way, when you look at the connectivity patterns observed in patients, going from fully vegetative, patients who have no evidence of any consciousness on the left side, to patients who are slightly conscious, or they're called minimally conscious, they present some sort of conscious behaviors, to patients who have a bit more evidence of consciousness. So there's kind of like a gradient, to all the way to the right, where are basically patients who are locked in. So these are patients who are fully conscious. They can even sometimes move a finger or something to show you that they're conscious, but otherwise they can they're paraplegic. They're not able to move their body, but we can tell that they're conscious. So you can see that the connectivity pattern starts to recover. So we can have even more sort of evidence that, yes, these patterns of connectivity must mean something, though we don't know exactly what they mean. But they mean something in terms of the fact that the stronger and the particular the pattern of connectivity observed in the EEG, the more conscious the patient is. But we can do more than just that. We can link these observed correlations in the connectivity of the brain to other forms of imaging, so multimodal imaging. So in this case, PET, which is positron emission tomography, which is measuring the blood flow in the brain. So patients, some of them show very strong amount of blood flow to the brain, right? Because essentially it's looking at the glucose metabolism of the brain. And compare that to patients who have very low glucose metabolism. So essentially that is an indirect measure of that usage of that brain area. And on the right, you have a typical, like a patient who has a lot of brain activity, and the patient on the left has relatively little. And if you compare the same group of patients in terms of their EEG connectivity, you can see a strong difference. Patients whose brain is demanding glucose for its activity, you see a lot of brain connectivity. So again, giving us more evidence that, yeah, these patterns of connectivity do mean something, and we can potentially use this in many ways. So it's good that essentially whenever somebody is conscious and whenever patients are conscious and are able to report that, we can basically build machine learning systems that can automatically take this connectivity pattern and try and predict, is this connectivity pattern coming from somebody who's conscious or somebody who's not conscious? And this is very useful. Why? Because in the clinical setting where these patients are living today, misdiagnosis is common. And that's because it's hard to know when you just observe these patients and their behaviors from the outside, is this person really conscious or not? Very difficult to know. So there is misdiagnosis where consciousness is missed. So what we have shown in previous research is patients who have an accurate diagnosis of being conscious have a pattern of connectivity that is strong, that is in the one in the middle. Patients who are accurately classified using neuroimaging other tools as being completely unconscious, using sort of careful behavioral assessment, who are classified as unconscious, are indeed showing no pattern of connectivity. That is very clear. That's on the left. But what's interesting is patients who have been misdiagnosed as unresponsive and completely vegetative. But actually, if you assess them again with a behavioral tool, like the coma recovery scale, and do it more systematically, and then say, okay, actually, just with the behavior itself, these patients actually are showing some consciousness. And then if you look at the EEG of those patients and the connectivity, you can see that they show strong patterns of connectivity. So these EEG tools can potentially be used to identify cases of misdiagnosis through sort of poor behavioral assessment, because not everywhere can you find experts who are expert neurologists to assess these patients, because they're quite rare and not all neurologists know how to assess these patients. So these tools can help that. Then finally, these tools are also interesting because they provide another interesting situation where they're useful. And this is in the covert cognition paradox. Now, I've got a picture of Roger Federer here. You might be wondering what's it got to do with tennis. And in fact, it has a lot to do with tennis. Because if I ask you to imagine playing tennis like Roger Federer versus imagining moving around the rooms of your house, what papers by my ex-postdoc supervisor at Adrian Owen showed was that there are clear patterns of activation observable in functional imaging when you play tennis, which is this supplementary motor area activation, versus when you're moving around the rooms of your house. So you're doing spatial navigation. A different set of areas are activating. Now, what Adrian did at the time, and it was considered crazy, was he asked what looked like a completely vegetative patient to do the same things, to imagine playing tennis or to move around the rooms of their house. And there were some patients, remarkably, who produced activation in pretty much exactly the same areas, though they appear to be completely unconscious. So the possibility that there might be hidden consciousness in some patients that is not evidenced in any behavioral assessment, however detailed, became a possibility. So what we've done since that original publication is shown that if you think about a healthy adult, and I think there are cases here, like in fact, like this picture of this one healthy adult, which was data collected in Chile through a collaboration there. So this might be a Chilean healthy adult whose brain shows a pattern of connectivity that is strong, and robust because they're conscious and they're awake and their brain is healthy. You compare that to a patient who is seemingly vegetative, and when you ask them to imagine playing tennis in an MRI scanner, you don't see any response in their fMRI activity. This is what their brain activity looks like. So confirming that, yeah, essentially everything indicates that there isn't a consciousness in this person's brain. Even covert consciousness does not exist. However, there are many patients we have found, these are not the majority, they are still a minority, but a significant minority of patients who appear to be unresponsive. There is no behavioral evidence of consciousness, but if you ask them to imagine playing tennis in the scanner, they produce the activity like the healthy adult would produce, and the EEG looks strikingly strong and really well connected in terms of the connectivity pattern. And remember, the EEG is much easier to do than putting somebody in an MRI scanner. So there is some value there as well. So hopefully, I've zoomed through a lot of data, many papers, but I've given you like a whirlwind tour of where functional connectivity can really be useful because it's easy to do in comparison to, let's say, functional imaging or something like PET, which costs a lot more money. And usually patients are not near a scanner and things like that. But as I said in the beginning, this is not the only way to measure and study consciousness in the brain. brain. There are other tools like DCM, so what's called effective connectivity. So let's take the same example, the sedation idea, which is easy to understand. You're taking somebody who's fully conscious and is a healthy adult, giving them an anesthetic like propofol, which is a very commonly used sedative in the hospital today. In fact, 30,000 people every day in the US undergo propofol sedation. And then you give them this drug, they become unconscious, and then they recover. And you can measure the change in their scalp or sort of EEG connectivity like this. But what if you took the same thing and you wanted to ask, with that data, some deeper questions about the nature of the connections in the brain that generate these patterns of connectivity, that generate the EEG you can observe. And that's where you can do things like dynamic causal modeling, which is a form of generative modeling of data. So what does this involve? So I'm going to take an example from our recent paper where we said, okay, we're going to look at a network in the brain. We looked at many different resting state networks, as they're called, but I'll just focus on one in the interest of time. So this default mode network, which is probably the most well-known resting state network of all, is basically a network that includes the prefrontal cortex, the precuneus, and the lateral prefrontal, so the lateral parietal areas on the left and the right. So it's essentially those nodes you can see there. So based on a lot of research in other areas using other modes of imaging, we know that when somebody is conscious and is kind of like in a sort of mind-wandering or self-referential mode of thinking in an MRI scanner, if you just put somebody into the scanner and say, just think about your day and your lunch and whatnot, they will basically generate activation in a set of areas that are shown in the picture here. So we know that if you're conscious, it's reasonable to assume that there is activation in these areas. And we can then say, well, what we don't know is, well, in any individual person, okay, these areas are involved, but how strong is the connection between these areas? And most importantly, when consciousness is taken away using an anesthetic, how does that connectivity change? So this is not something you can answer with sort of functional connectivity at the scalp level, because it's difficult to know exactly what's going on unless you do something like source reconstruction. So here with DCM, instead what you do is you say, well, let me assume a certain specific network like this, like shown in the picture on the right side. That's a functional kind of architecture of the network that I'm proposing. What I want to know is, what are the strengths of those lines, the dashed lines and the dotted lines, which are the strength of the connectivity network. And you can do the same thing as we've done in this paper for different sorts of resting state networks, like the salience network, the central executive network. So you can basically construct these networks first and then say, okay, now I also want to ask about how the connectivity between all of these networks is also changing, so inter-network connectivity. So you see, you can start doing some pretty funky, complicated modeling where you've got loads of nodes and lots of connections between these networks and ask, I really want to infer the unknowns here, which are the strengths of the connections between these areas. And so zooming through a lot of theory around how DCM does this, and sort of saying, well, let's leave that for another day. What we can do, the long story short, is you can infer, exactly as I said, the strengths of those connections, more specifically, how the strengths are changing when somebody goes from being conscious to unconscious. So a graphical representation of that is on the left side to make it easier to process. But essentially, the long story short is what we're showing is that there are strong changes in the strength of the connectivity between specific areas, especially in the posterior and frontoparietal connections, when somebody goes from being conscious to unconscious. Now, this might sort of really put you in mind of theories like the hot zone, theories that, Tononi and others have posited that there is a posterior hot zone in the brain that is basically involved in sort of maintaining consciousness, as it were. And of course, this relates to what Andres has talked to you about in terms of the connections that seem to change in scalp level data when the contents of consciousness are changing and so on. So essentially, what DCM is trying to do is sort of pin down quite specifically which connections are changing, how much are they changing, etc. So in the paper, we sort of do a detailed analysis of all the different networks, and I'd invite you to have a look at that to sort of understand, can we sort of identify to the extent to which hot zone theories have some value. And this is where DCM really becomes useful, because we can take what's a textual theory written in some paper in words and try and make it quantitative, put some numbers to it. because the theory of the hot zone is fine, but exactly how hot is hot and what bits are hot and what bits are cold is difficult to sort of make any claims based on text. But if you can actually model it, you can sort of say something more specific and try and disambiguate between different possible postulates you can create based on a textual theory. Okay, so the last sort of thing I'll say before I end my talk is out-of-sample prediction, and this is where it becomes even more interesting. What you can do with tools like DCM, and I mean, you can do this with functional connectivity as well, is you can try and do something that is very useful to understand how general these findings are. Findings like, well, these connections are changing like this or that. I mean, is that really true, or is it just like some sort of artifact or overfitting to the specific data set I have? Now, this is a very important question all modelers should ask. Is my modeling robust? And there are many ways to do this, but one particular which I really like is to use something called out-of-sample analysis. So you can take data from 10 participants you've collected data for, and then, as I said, go from not knowing the strength of these connections to inferring the strength of those connections using the data, and that is called parameter estimation. So going from the observed data to the unknown states of the brain. And then you can take that unknown state of the brain and try and predict what data you would expect to observe for the 11th participant. This is the power of generative modeling. Because you can generate data, you can basically generate new data for a new unknown participant, and then try and say, well, is that prediction good or not? That's what we did in this paper. This out-of-sample prediction, and you're seeing the kind of summary of the results here. There's a lot of stuff in this figure that I don't need to go into. All I want to draw your attention to is the fact that you can basically make some specific claims that you can't do with sort of scalp level functional connectivity analysis, which is, as I said, strictly correlational. So from the scalp data, you can't say anything about specific regions in the brain. But here, we can do that. We're saying it appears that when consciousness changes, the frontoparietal connections, the DMN, seem to be changing a lot. And more specifically, the strength of the change in the frontoparietal connections explains the change in consciousness in the 11th participant, the new data set. So out of sample, basically, put it in plain English, you give me some EEG data and I will use the frontoparietal connections in that data to predict whether this person is conscious or not conscious. Right? So that sort of is basically predicting new data, which is really powerful. So in summary, I think I'm quite well for time here. I wanted to highlight some key top-level stories that hopefully I'll try to convince you about. Firstly, that at a high level, there are many different tools that are kind of increasingly popular in neuroscience, especially with the increasing use of computers and sort of computational methods in the field for using functional and effective connectivity analyses to sort of ask deeper questions. And this has not always been easy because of the lack of tools. And over the last 5 to 10 years, I've seen a dramatic increase in the availability of such tools and the ease of use. And these are all valuable and have, in many cases, not only value for understanding, but also applying it for clinical usage. And I'm happy to say that the functional connectivity example I showed you with those Mohawk patterns is today used for assessing patients in the clinic in multiple centers in Europe, where they're using it alongside other imaging techniques to try and get an index of if they get patients who are being assessed at a hospital, can we do some EEG on them to get a quick idea of whether this patient has some evidence of consciousness or not. So there are examples in neurophysiology as well. For example, to study or measure the depth of anesthesia during a clinical surgery, because it's hard to know in many cases whether a patient undergoing surgery is truly unconscious before they begin the surgery. And there are cases of patients feeling that they're woken up and they can feel the doctor doing stuff to them and they can't express themselves because they're paralyzed. But they then report post hoc that they felt conscious. So obviously you don't want that. So this can help in situations like that as well. And of course, the neuropathology example is the one with patients. And there are other examples and papers that are applying these sorts of things in psychiatry and other sort of subfields of medicine as well. But I will highlight that none of these methods is perfect in any way. They all have advantages and disadvantages, and there is no perfect method that can truly tell you what's going on inside the brain, because to have that would be, as they say, to know the mind of God type thing. We don't quite 100% have such a tool. And hence, all these things can do is complement insights from other sources of information, like structural connectivity and other modes of imaging. So that what we're looking for is convergent evidence across multiple domains to give us real confidence that we're on the right track. So on the whole, the key point there is that it's all like this kind of Plato's cave analogy from thousands of years ago. We're only looking at shadows of things when we measure the brain. And from those shadows, we want to infer the truth. It's always going to be limited by the fact that shadows are fundamentally imperfect representations of the truth. We should always keep that in mind as modelers. Never claim that my model is the best model or the correct model. As somebody famous once said, all models are wrong. Some models are more useful. Okay, with that, I'd like to thank all my collaborators. This is an incomplete list, as always, and all the funders who funded many aspects of this research. And thank you for your attention.
Well, I am learning so many things today. Thank you, Srivas, for your great presentation. I literally love these punkish connectivity pictures. I love them. And now it's time to present the next talk. Cees van Leeuwen is a professor and leading researcher of the Brain and Cognition Research Unit at the University of Leuven, Belgium. I had the pleasure to meet him personally before the pandemics in many conferences organized by the Sapienza University of Rome, which is my alma mater university, since I come from there. So I'm really thankful that he accepted my invitation to be part of this symposium today. He's interested in research topics as visual perception, consciousness, complex adaptive systems, and brain dynamics in general. He is also the head of the Laboratory of Perceptual Dynamics, where visual awareness is studied using a complex system view, unraveling the temporal structure of visual experience and its neural underpinning. Today, he will tell us about structural and functional connectivity, a simple tale of optimization. Cees, the lights are on for you, please.
Okay, so structural and functional connectivity are the topic, and the topic is actually very old. At least in my lab, we have been working on the relation between structural and functional connectivity for a long time, since about 2000. That's more than 20 years now. While at RIKEN, and I would like to mention the main actors there: Poon Gong, Daan van den Berg, Hoi Fei, Antonino Lafone, of course, and Mikhail Rubinov. And then at the KU Leuven, where I transferred in 2011, Nick Jarman, Carlos Calvo Tapia, Manuel Hakiat, Ilias Rentsepiers, and most recently, Jia Li. So these people have been engaging in modeling and providing a generative model that expresses some important and interesting features of the relationship between brain structure and function. To give you an overview of the talk, this is the introduction. I'll talk a little bit about complexity and the basic ideas that we introduce in our models, which is the notion of adaptive rewiring. Now, adaptive rewiring, as I said, has a long history. We started out modeling these things in 2000, and there are probably 3 different stages. The first 10 years, while still at RIKEN, the second 10 years, while in Leuven, and the future. And the future, as we all know, is uncertain. And then some concluding remarks. First of all, brain anatomy can be regarded as a complex network. Yeah. What do we mean when we say complex network? Well, we talk about the way different cells, different units of larger scale at the brain are connected and form a structure. Yeah. And you can have connectivity diagrams, and these connectivity diagrams show all kinds of interesting features, such as some degree of clustering here and there. Also notice how relatively sparse they are. That means that if we can reach good connectivity and good communication in the in such networks, good communication between signals in such networks, That means that the network connectivity structure must be highly efficient. Yeah. So this is not just at the level of brain. This is another. As I said, the brain structure is complex and efficient, and these features of complexity and efficiency can be translated in more technical terms in properties of the neural network graph, such as small worldness, modularity, and rich club. Small world is typically something like the best of both worlds between a regular network and a random network. As you can see, a regular network has optimal local connectivity. You can get from here to here very easily and from here to here. Yeah? But global connectivity takes a lot of intermediate steps. Yeah? A random network has optimal or good global connectivity, but there is no local clustering, so communication between local parts of the network is difficult. Now, a small world network is basically clustered, but with very strategically added global connectivity. And it turns out that the very sparse global connectivity is sufficient to reach a high degree of effectivity in the network. So that's one feature of networks that we think is contributing to the high integratedness of our brain. Another one is modularity. Modularity is the fact that there may be isolated or relatively isolated brain regions, but these are always connected through a network of so-called hub nodes and hub connections, which means that there is both segregation and integration possible in the brain. These modules can work separately and independently, but if the connection, the global connectivity is optimal, they can also work together and integrate their information in an optimal way. And finally, this optimality is maximally ensured if these hubs have a rich club structure. That means if the hubs themselves are all interconnected, and then we speak of a rich club. So this is the optimal design, you could say, for a brain, but you cannot design a brain like that. The brain we know has about 20 billion neurons. All these neurons have maybe between 5 and 20,000 connections, so that's a lot. You cannot encode it into genes. You need to somehow be able to obtain that epigenetically in some way. Yeah? And therefore, the million-dollar question is, where does this complex topology come from? And well, this question can be studied in great detail with great neural realism and with lots and lots of sophistication. We, however, don't have that firepower, and we are not also that much interested in too realistic efforts to model the brain and to model the brain generation in the current stage, where still so much is unknown about brain structure and function. So we need an easy and low budget answer. Yeah, and my low budget answer is adaptive self-organization. And what do we mean by this? Well, adaptive self-organization implies that the brain is always shaped by its own spontaneous activity. Yeah? And we see that in the unborn infant, we see it in neonatal rats and mice. And But we also see it later in the development, and we think that this is a continuously ongoing process. Spontaneous activity helps shape and optimize the neuroanatomy, which in turn maximally supports the efficiency of the spontaneous activity. So this is why we speak of this too as a synergy. Now, we have two ingredients here: spontaneous activity on the one hand and neuroanatomy on the other. And the plan is to make a model of this, which is as simple and as abstract as we possibly can, in order to find some principles, some very simple basic features that characterize this synergy. Yeah? So how do we go about? Well, first of all, let's look at some spontaneous activity. Here I say. All right, so here you're looking at spontaneous activity in EEG phase space. You're viewing the top of the head, the nose is on top, and what we see is that this activity is highly structured, and that there's a lot of dynamics. There is traveling and standing waves. The traveling waves go in all possible directions, from front to back, from back to front. They also make circular patterns, and they alternate, and they are typically not stable. They don't stay forever. One appears, then there's a brief period of instability, and then another one appears. This is a typical characteristic of spontaneous brain activity. Right? And well, model this is a very already very simple neural mass model, which I will not go into detail, but this is even too complicated for us. We make it even more simple. We do a cross section through this return map to get a sort of one hump map here, and we simplify this even further. So this is what our activation function will look like. Yeah, and you've seen that the previous one was noisy and this one is not. But if you have these maps coupled, then they will perturb each other, and therefore there will be some appearance of noise nevertheless. Yeah. So we couple them in the way a brain is coupled, a small world. Yeah, and we will see what this delivers you in terms of activity patterns. You see that there is at least a qualitative resemblance between what you've seen in the spontaneous activity, traveling and standing waves, alternating periods of metastability, so to speak, where certain patterns prevail for a certain amount of time and then collapse, and something else will come in its place. So this is it seems very much a generic process as soon as you couple nonlinear oscillators. And it has to be, because it is so basic. And if it's so basic, then it is something that perhaps is generic. And if it's generic, then evolution can use it. Yeah? And this is why we think that this type of activity patterns are actually employed in the brain to optimize the structure. So the question is then, how does the brain optimize itself in adaptation to this structure? Well, in adaptation to this activity. Well, we think if we consider neuroanatomy as a graph, then there may be some initial design, yeah, but there is some way to enhance this design by just making the communication more efficient. Yeah? And that means that this path will over time be obliterated, while this path over time will be strengthened. And this, of course, can be very abstractly modeled by this process, which is called adaptive rewiring, which you can see here. If between two units of a network there is intense communication, you add a connection there. And if there is no communication, that path will be obliterated. And this is how we think that we can achieve this optimization gradually over time, just adaptively rewiring the network according to the communication patterns in the network. For instance, the synchrony that occurs between two unit nodes. So this is how we started out in 2000, and in the past, this is how we initially modeled this approach. With this approach, we had oscillatory activity patterns in coupled maps, just as you have seen, and we do the rewiring based on synchronization. That is, if there is synchronization between an uncoupled pair of units, we add a connection and we cut the connection somewhere else. And when you do that, you get this. What you see is now that an initially random network is gradually adaptively rewiring itself over time according to the patterns of synchronization that occur in there, and that the structure is becoming increasingly interesting. So what we end up with is connected clusters of units and globally connecting hubs. In other words, and of course these hubs are interconnected. So lo and behold, this system that arises here has all the properties of a small world network, modular, and rich club structure. So this is what we can see in this very simple model. Here are some publications that attest to that. Okay, now from this very first beginning, we developed some nuggets of wisdom. One observation that was made by Rubinov in 2009 is what exactly is the relation between the activity patterns and the structure. What follows? How does the functional connectivity precisely affect the structure? Well, it turns out that the patterns that you see from moment to moment in these fastly changing oscillators are not so relevant in shaping the structure. It is rather more the moving average of the activity that shapes the structure. And that is becoming important in the next stage. There are also some specifics about network pruning and spatial embedding. I don't know if I have time to talk about this, but pruning is, I think, interesting. If you take a network that is sparse, then it will actually create this small world structure. But if you cut connections such that it goes beyond a critical threshold, then the system will no longer be able to generate this typical small world structure. At the threshold of connectivity, there will be some kind of efforts being made. The system will go towards the small worldness, reach a platform, and then collapse. As you see here, it grows again, reaches a platform and then collapse. Yeah. So this is the connectivity that is exactly at the critical level for the growth of the small world structure. Yeah. So you see here that this is the growth of the small worldness. This happens when the connectivity is just too sparse, and here we see the event when the connectivity is at the critical level, just not enough to obtain this small worldness. Yeah, and you see that it grows in complexity, reaches a platform, and goes down again, and does that repeatedly. Yeah. So this is a typical phenomenon that we see in, for instance, all kinds of developmental diseases such as schizophrenia, where there is an intermittency, a hopeful development plus a collapse, a hopeful development plus a collapse. Yeah, this is a typical pattern. So we think that this could offer a way, we thought that this could offer a way to, an interesting way to model these developmental disorders. Okay. So another interesting thing is that, of course, this is a graph model. There is no such a thing like distance in the model other than the number of connections you traverse in order to get from one place to another. But you can, of course, also introduce distance, physical distance, to such a model by putting it into spatial embedding. For instance, you can project your network onto a ball or a cylinder, and when you do that, then there is also a physical distance between nodes, yeah? And then the physical distance may start to matter. For instance, if you do a rewiring, which is between two units at a large distance, that will come at a cost because you need to make a long connection. Yeah? So if you want to avoid this, you can use the rewiring in combination with some algorithm that penalizes distance. And then the system not only minimizes the topological, maximizes the topological efficiency, but it also minimizes wiring length, and it finds some kind of optimum between those two features. You can see what happens in that case. You will see that if there are no spatial constraints, then this model will just have no relationship whatsoever between the status of the nodes and the distance and the length of the connection. Yeah. So if the connections are hub connections or not, is completely unrelated to how far away, how far removed these two nodes are from each other. Yeah. So, but if you penalize the distance in different ways, then you see an interesting relation appear. So in fact, you can see this here, a correlation. Here's the spatial distance, and here is the betweenness of the edges. And what you see is that the edges that have the small betweenness, that is the local connections, yeah, the ones within, you could say, network clusters, they all have a small spatial distance, or tend to have a small spatial distance. And the ones that have a high betweenness, in other words, the hub connections, have a large spatial distance. In other words, this network, if you compromise between evolving an optimal network and in terms of topology and in terms of space, develops automatically a structure of hubs where the within hub connections are all spatially local and the between hub connections are spatially long range. Yeah? So you get something like a topographical map. And of course, depending on how you weigh exactly this distance, you can tweak the system in interesting ways. For instance, here we see that modules are relatively nicely segregated, but the system is not completely clean in its segregation. You see some connections between different modules appearing where this one, for instance, belongs to the pink module, but is situated into the green module. That means that there is some overlap, or some you could say some interplay between the modules possible. And that's a very interesting idea, because this degree of interference can vary with the character of the weighting. Here, for instance, you see a different weighting of spatial costs, which results in a far more messy modular structure. Now, this has advantages and disadvantages, and so does this. This, for instance, suffers from heavy interference between processes within modules, but also easily makes long-range connections. Yeah? So this type of network, we could say, relatively to this one, is dyslexic, but it's also creative. This one, we could say, yeah, in the extreme case, is autistic. Yeah? So we could say that by tweaking the different weightings of the distance and topological optimization, we can get different types of behavior, both within a certain network and for the, yeah, you could say the model brain as a whole. So let me quickly say something about the present. We've already said that the adaptation to the activity follows the, you could say the long range or average activity patterns, not the short range ones. It means that the oscillations are actually not so important, and therefore we can simplify the model quite substantially. So instead of having activity based on oscillations, we're now looking at activity modeled by simply diffusion, and that's far easier because we are looking now at processes that are taking place in a stochastic way over time in our models, and let the activity flow guide the rewiring of the network in a much more simple way. I have much more, which I will not be able to tell you, but you can at least see some pictures. Again, there are issues of balancing the different ways of tweaking the networks through. Here you have an example where you can have both predominantly modularity or predominantly centralization, but we can also find a critical equilibrium here, where we have equal amounts of modularity and centralization. And in this type of networks, we recognize the connectivity that was presented initially, for instance, in the mouse brain. So what we see here is networks that typically have the hierarchical structure of the brain. can do something with space here, which is now far nicer because we have far better grip on the spatial principles of wiring distance, for instance. And you can see that we can do quite well also on other things that are relating to alignment between neural connections, simply by following this diffusion idea. And what you get then is you get all kinds of interesting spatial patterns, which are the stalwarts of the of simpler small brain networks. Finally, the future, and here I would just point to one thing. All the networks we have presented so far are undirected. I said we are aiming for the simplest possible models. And if you talk about synchrony in brains, then synchrony is typically happening in an undirected fashion. A network unit A is connected to B just by synchrony, just as B is connected by synchrony to A. However, diffusion allows for models to be directed, that is, diffusion can go at different rates in one direction or with different preferences from one unit to another or back. Yeah? That means that we have an asymmetry now at our disposal, but we didn't have before. And depending on how we value this this diffusion direction, you can get two types of hubs, namely input hubs and output hubs. And the interesting thing is, if you get both of these in your networks, you have so-called convergent divergent units. Convergent divergent units are ones that consist of a convergent hub at the beginning, some intermediate nodes in between, and divergent hubs at the end. These kind of structures are optimally suitable for context-specific processing. Therefore, we tried and succeeded in modeling these units. Okay. Some publications on this. I will end by saying what we achieved is showing that adaptive rewiring offers a versatile principle to elucidate how brain connectivity structures could emerge. It is a just so story, we know this, of course. We also shown how this could interact constructively with spatial principles. And our models are generative models, which means that they have a number of testable and tested consequences. We know about the complexity, we know about variant brains. And when I say variant brains, we could also talk about variant brain areas or differences between. We have the convergent divergent units. And last but not least, we have the global workspace.
Thank you for your presentation. Usually your presentation was very interesting and clear. So I hope that now we can have some time to answer the questions from the public. So I will check if there are some new. No. So now this is the Q&A session. I will read the answer from the questions from the public. And then if, of course, if you have also the other speakers, we can address questions each other, I mean, amongst ourselves. So I will first read, I think that the first were for Andres. So the first one is, in the brain perception, is the fact that something we see is animated versus a still image playing a role? For example, the two faces vase versus the silhouette dancer rotating left or right depending on our perception. Also, is the 3D versus the 2D playing a role in the final outcome of our perception, or we perceive both 3D or 2D in the same way in terms of cognitive processes? So this is for Andres.
Okay, I actually don't know, but if I have to say something meaningful, I would say that, so I think the first thing that we need to distinguish is, I would call symmetrical versus non-symmetrical kind of illusions. Because like the Necker cube, for instance, the classic Necker cube that you can see either the front of, or the back of the cube, I would say that's more like a symmetrical one, because at the end of the day, what you're seeing is different angles of the same cube. So it's still perceived as a unitary object. But there are other illusions like the ones that I showed that are non-symmetrical in the sense that you either see one object, like the case of the vase face one, or two. So basically it's one person versus two. And the auditory illusion that I show you is also non-symmetrical in the sense that you can perceive one or two objects. So I would make that distinction first between this kind of symmetrical versus asymmetrical illusions. And I think the second part of the question was whether the brain is processing in the same manner illusions that are related to movement and 2D versus 3D. I would say that, of course, that's a priori, I would say that that's the case. I don't really know whether we would see the same kind of neural markers that I showed using this kind of illusions. What makes me happy is that when you test two different modalities, like the auditory modality and the visual modality, we observe the same effect in terms of neural metrics when you press buttons. So you see that the integrated percept, both auditorily and visually, is basically correlates with an increase in neural integration, and the same with the auditory and visual differentiated percept. You see an increase in differentiation before people press the button. So at least we have that. These two examples that might be completely different. One is an illusion that is generated by the motion of the grids, and the other one is just an illusion that happens over time because of the tones. And still, even though they're very different, you have the same kind of mapping in the brain. So maybe there is some kind of underlying general kind of mechanism by which these illusions are generated. I don't know. At least for now, we have these two examples. So it would be nice to. So thank you for the question. It would be nice to actually check whether other illusions, 3D or with other kinds of movements, are actually reflecting the same underlying brain processes. So that's what I can say for now.
Thank you, Andres. You have another question, actually. So first, congratulations, nice presentation and research. What conception of consciousness are you using in your research? In precise terms, how would consciousness differ from the mechanism of earlier perception, mental imagery in the first case of your research, and the attentional switch that functions as a figure and ground operator in the second case? I mean, what are the differences in the study of neural correlates of perception and attentional switch and the neural correlates of consciousness?
Thank you for the question. That's a very good question. I think what I'm referring by consciousness is about the experience of the participants. So basically, when I use the word consciousness, I'm not referring to any kind of brain process, even though I'm using brain activity to understand what happens with it, but I'm genuinely, I'm actually referring to the experience. So when I use the word consciousness, I'm referring to what people are actually seeing, actually hearing, actually touching. And in this case, that's why I'm very, I said that these concepts of integration and differentiation are very useful, because phenomenologically speaking, from a first perspective point of view, we can kind of make certain claims that are generalizable across people in terms of how they perceive. So there is an experience of seeing wholeness in the world, even though you can make distinctions. So if I have this image in front of myself now with these two screens and your faces, I can clearly say that there is a unitary experience, even though I can make distinctions within that unitary experience, which is the face of Pedro, the face of the screen, etc., etc. So my definition of consciousness is phenomenological, is based on experience. That it's a very different thing than starting from the brain. And I think that's a major confound that we might have in some cases, that we start defining consciousness as something or some neural process that the brains do, right? Instead of starting from experience and then tracking back to the neural markers. So I'm trying not to do that. I'm trying to start from an experience that everybody can feel, in this case, a transition between one object or two into modalities, and then tracking back to the brain. Having said that, of course, when you experience something, there is attention allocation and there is working memory involved, and there are all of these cognitive functions that we know. But I'm not referring to those. I'm mainly referring to the ability to experience certain things. And that's why I understand by consciousness.
Yes, thanks for your answer. Then I have a question that I don't know to whom, but maybe you can all answer if somebody has an answer. The question is there a characteristic brain network for metacognitive thoughts? Maybe maybe it's about more about metacognition. Yeah. Who wants to answer this question?
I don't know. That's my answer. I think there is no.
Some.
Yes. Chess?
Chie will talk about certain aspects of cognition that have a metacognitive component to it. Maybe you could pose the question to her later.
Yeah. Yes. I think that this is metacognition is more like composite. No, there are no brain networks for that. Yes, the questions from the public are finished. I didn't.
Can I ask a question?
Of course. Now it's our turn, so we can ask questions to each other.
I have a question for Srivas actually, because I haven't seen the last results that you showed. So if I understood well, when you do this one out patient thing with the modeling, what you said is that if I train or if I look for markers of, in this case, of consciousness and understood as different levels of, arousal, and I try to find them in a bunch of people that I know, the idea is that if I give you a subject that you haven't seen, that the classifier or any technique that you're using hasn't seen at all, can I use the information from these other patients in order to tell whether this is or not, let's say, a vegetative patient or a comatose patient or a minimally conscious one? Did I understand that correctly?
That's exactly right. So you're asking whether you can do this sort of out of sample thing. Given a new data set, can you predict essentially the likelihood of certain inner states being true or not? And what we show in that paper is that not only can you do that, you can say, well, what is the predictive power when you're trying to do that, when you use this connection or that connection as the predictive marker, right? And what I showed in that figure was that the frontoparietal connections provide the strongest predictive markers for predicting whether a patient, and that focus in that particular paper was on healthy volunteers who are undergoing anesthesia. But in the thesis of my PhD student who's done all this work, he has a section where we use the same technique, but for patients, as you say, try and predict based on the specific connection inside the brain rather than at the scalp level, can we predict whether this patient is conscious or not? And we use that to show that the patients who are covertly conscious, who play tennis but look unresponsive, they have very strong connections in between those specific brain areas, which can be inferred through DCM, fitting with convergent essentially. This is not saying anything that could not have been guessed at, but guessing is one thing, proving is another. So just converging to the fact that at the scalp level we see frontoparietal connections. Your work shows similar ideas, but this is showing that you can basically use generative modeling to identify specific connections and ask how much does it matter related to other connections. It brings a more quantitative way to be able to do out-of-sample prediction.
Mm-hmm. And those predictions are like dichotomous, so basically is it a patient or is it not? Or you can also have an estimation of how good, if you basically have more than two categories, if you have vegetative, comatose, minimally conscious, etc. Does it work?
That's a good question. Yes, you can do that, exactly. And what it gives you is the probability of group affiliation. The probability that a particular data set belongs to one of many groups. So just like a classifier, just like a machine learning system. It's fundamentally no different. It's just that it's working off of a different mathematical substrate, which is probability distributions, other than a hyperplane or a deep learning trained model. There are some distinctions that I won't go into, but it's essentially producing the same functional equivalence in terms of what it can be used for. It's just more computationally demanding to build and train a generative model.
Thank you.
Meanwhile, I got another question from the public, this time for Cees van Leeuwen. What do you think about some perspectives that holds that higher cognition are based on basic perceptual and motor processes and that this is a problem for modular perspective? Does modularity apply only to basic cognitive processes, or could we conciliate a modular view for higher processes too?
This is a very interesting question, because hello, microphone on?
Yes, we can hear you and
Okay,
we can
great.
see you.
Thank you. So I think this is a very interesting question, because the notion of modularity and encapsulation versus global connectivity has been put in opposition in a way that is rather simplistic. Yeah? In the models that we have, as you can see, we have all kinds of in-betweens between modularly connected and centrally connected. Yeah? And we can tweak these as a continuity and find an optimum point for a certain function. Yeah? So it is neither central processes nor sensory processes are exclusively modular, because the brain is interactive, but it interacts in different modes, and these modes sometimes follow a more let's say modular, isolated fashion, if you like, and sometimes they are more integrated. And I think we have seen some nice evidence of this in the earlier presentation. Yes,
thank you for answering this question. I also have a question for Cees van Leeuwen. Well, you have models, as you say, they were like limited because of the complexity that you can reach. I have the feeling that many perceptual processes or whatever process the brain can manage are attained to the way of functioning of the brain. So each brain works in some way, has a certain optimization, has a certain distance from hubs and so on. So in a global way, you can say this brain pertains to this person, so you can somehow go back and understand how this person works and functions. But do you think, or would you say that sooner or later we will be able to have all these configurations of, for example, registering EEG from many persons and, I don't know, train some machine learning algorithms to distinguish between certain states and so on, and then it will be easier in the future to make like diagnostic processes also and so on?
Well, I'm not so sure. First of all, I need to say that what we are doing is we are talking about how brain structure is shaped by activity, yeah? And the fact that we can show how brains more or less are optimized for processing does not specify the details of how these processes actually occur. They just parameterize these processes. We say, well, certain processes may go better in a more modular fashion, others may go better in a more integral fashion. Some are good when the modules are more context sensitive, some are better when the modules are less context sensitive, and so on and so on. So these are the things that we can optimize for a brain. And then the brain is the playground for an information process. Doesn't mean that we actually model the information process. How the information process actually runs on such a brain is a different and rather independent story, because we just give the brain the freedom to do it in the best possible way. Doesn't mean that it's also always done in that way. Yeah? And probably and possibly it is done in different ways at different times. We have a lot of variability, and we see this variability in trial-to-trial EEG, for instance. Yeah? This variability is so large that we probably have very little to diagnose if we just take the, let's say, the recordings of brain activity and say that this is how we're going to then decide how somebody processes information. I think there is too much variability in that, both in the neural signal and in the way the processes run.
Okay, thank you a lot for answering. If there are no other questions, then we can make a small break and then pass to the second session of the symposium. Here we are again, ready for the second part and the last 3 talks for today. I will shortly and briefly introduce Chie Nakatani, the next speaker. So as the previous speaker, Cees van Leeuwen, also Chie Nakatani is part of the Brain and Cognition Research Unit at the University of Leuven in Belgium. She's a postdoctoral researcher in the Laboratory for Perceptual Dynamics, University of Leuven, Belgium. Before that, she was researching in the Riken Brain Science Institute in Japan. Her topics are dynamics of visual perception, neurocorrelates of temporal attention, transacaric integration of visual information. And today she accepted to be here with us. I'm very grateful for that. She will present and talk us about a dynamic scenario towards mind wandering. So Chie, the stage is yours.
Today my talk is most of the side of dynamics of consciousness. This is one of the Chiara's visualist. And this is a relatively new study, which I do with my master's students in case. So any suggestions, advice are really welcome. Okay, so let's get started. I'm going to talk about mind wandering. So mind wandering, so we experience it on and off. Our mental focus is somewhat drifting away from where we're supposed to do and somewhere else. So it's kind of a dynamic process. Yeah, so it's not just a state, but so how we actually get out from the focus and then wandering around and coming back. The other thing is which is not said explicitly often, but tacitly assumed is if you are drifting away from what we are supposed to do to somewhere else, which means there are more than one thing in your brain, in your mind. So there are multiple thoughts in your mind. So which means quite a bit of spontaneous thoughts. Thoughts are generated spontaneously. So spontaneous thought generation is the other side of one of the important aspects of mind wandering. So I'm going to therefore talk about control side and then thought generation side throughout this study. Okay. So here is me. So sometimes I'm really focused and then I'm doing everything right, focused. But at other times I'm just mind wandering. But one more other time, so I'm juggling with many things in my mind, but I'm completely in control. So maybe I can sort of give some sort of structure by using two dimensions. Is, as I said, number of thoughts in your mind. So how many thoughts in your mind? Large number or small number? And then the other dimension is the level of cognitive control. Do you have weak control or quite strong control? So and you actually see a blank part, so which is cognitive control is low, and then number of thoughts is low. So what could it be? That could be some kind of obsessive thoughts and stuff like this. So that somehow which actually this very simple scheme holds quite varieties of spontaneous mental state. But what I am interested in, not kind of state, but which we actually are sometimes is a multitasking and just doing some task great and the mind wandering and so forth. And we experience those different states and we can describe it as a trajectory. Yeah. So in a more abstract manner, so we can think of mind wandering. So as a part of large varieties of spontaneous thought process. And to the point, well, let's say from single task focus condition, are we going straight away or going somewhere else and so forth? Yeah. So as a matter of fact, we can think of infinite number of trajectories. Then, so how? But how does it look like in the real world? So I'm an experimentalist, so I just thought, okay, maybe I'll just do some kind of simple study. That's what I did with my master's student Ben Ganshaw, and then for that I used the real life sampling method. So that is fancy name, but it's just a question survey task done by the cell phone. So we send participants' cell phone a probe. The probe is a mixture of questionnaires, which asks for a number of thoughts or level of mind wandering and other experiential aspects of the mind at the moment when the participant is received the query, and that also the visual task, which is to measure level of cognitive control. So this whole thing takes less than 5 minutes, and then we send the 5 probes in, say, so variant intervals and 5 times a day, and we actually repeat it in 2 weeks. So based on the data, so the data we collected, analyzed from the time point of the report of mind wandering. Okay. So how the data look like? This is the results, and this is the level of cognitive control, high, low, and then this is the number of thoughts. Large number, high number, low number. And then this is a time point when the mind wandering is reported. So as you expect, the level of cognitive control is very low when a person reports mind wandering, and then after that they actually recover slowly and so on. So by the way, this is minus 8 hours plus 8 hours. This is quite long duration, which covers the awake time of the participant. Okay. And the number of thoughts are a little bit more, I can say, sort of oscillating. So it goes up and down. Up, of course, at the moment of mind wandering, it is high. But you notice there even already the 1 hour before the mind wandering is reported, the level of the number of thoughts are very high and go down and again. So in a way that actually oscillates, 4 to 6 hour cycle that's in a ultradian range. Okay. So they are not exactly the same, but they are somewhat related. So how we can see it? So now we want to see it in the form of a trajectory, like we as we planned, we have one number of thoughts as one axis and cognitive level, cognitive control as for the other axis. So how it looks like, this is a trajectory towards and then from mind wandering. This is the point of mind wandering. So let's start. This is 8 hours before the mind wandering. We start from here, which is cognitive control is high, and the number of thoughts is sort of moderate. And from that, we actually went up to some kind of multitasking regime, and then goes back to sort of single task, and then go back to the multitasking regime. So you notice that the number of thoughts is high. From here, we quickly lose the cognitive control to mind wandering. And after that, we dwell in this vision a little bit and then escape from the vision. So this is the trajectory of mind wandering. Okay, so that's not very simple straight line from single task to mind wandering. So I can sort of say a mind wandering process unfolds. So in n-dimensional space, I say n, I use 2, but it could be more than that or less than the 1 dimension, so it's possible, say, but n-dimensional space. And then it actually develops in real time. Yeah. And then this we can sort of study it as trajectory. Okay. So those things are quite different from what at least I'm used to, which is a conditional means. We just take a mind wandering state versus a non-mind wandering state of some sort, just to compare as a conditional means. So those things that particularly doesn't tell us anything about those dynamic aspects of a mind wandering process. So but for this top sort of presentation is quite offers interesting possibilities. Okay, then what I can say is next. Right, so now we have nice trajectories, but now what we want to know is how the brain is, or neural activity is modulating those trajectory. How should we think about it? So we have two cognitive functions, cognitive control and spontaneous thought generation, which is important for mind wandering. And then, so neuroscience communities already propose varieties of neural networks, these things, but maybe we can just very roughly classify them in two groups. One is the networks, which is for goal-directed behavior. So we actually call it goal-directed networks. And then the other is the opposite, so to speak, default mode networks. And then maybe we sort of think about some kind of modulation like this. So GDN, goal-directed neural networks, they facilitate cognitive control, but they inhibit spontaneous thought generation. But in the opposite to the default mode network facilitates spontaneous thought generation, but they sort of inhibit cognitive control. So this type of very simple but the antagonistic network would predict a timeline, time course like this. So when spontaneous thought is high, cognitive control is low. This is mind wandering. And the opposite is when cognitive control is high and spontaneous thought is low, you are task focused and so forth. Yeah, okay, this is a very simple idea, but often you see in the introduction or discussion of many papers in the field. But I have a little bit of problem here because if we actually support this type of immediate antagonistic model, mediator, then the all activities are some of the staying on the anti-diagonal line. The reality is not. So in reality, we get the trajectory like this, meaning that the modulation that we actually think about is probably not that simple. And maybe the timing of the say modulation could be variable, or even the direction of modulation can be variable. So we some more neural effects with variable timing and over direction. So we can probably think about, well, if I borrow the term from engineers, we can think about some sort of transfer function between these two networks, neural networks and psychological functions. Yeah, so it's good, but is that really so? Do we actually have some kind of neural effects which is variable in timing or variable in direction? So then, let's test it. So I did an easy experiment. Yeah, this is done with the Hannah Bernard. That's in the task. The participants a very boring task, which is called tone counting task. At the beginning of the task, this is the auditory task. Yeah, then at the beginning of the task, they are given a number, count n tones, n could be 20. Then a tone is played, beep, 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 and you actually count 1, 2, 3. And then if it comes to 20, you press a button. And from this, you get the correctness of the count. And then also the participants are supposed to report the number of thoughts that they had during this very boring task. And then also the percentage of mind wandering. At the same time, we record EEG. And then from the record, we compute the auditory evoked potential and then the alpha band activity. Yeah, this is, so to speak, the measure of a goal-directed activity, and alpha band activity is a measure of default mode type of activity. Okay. So from this, we get indeed 5 time series of data. One is the percent mind wandering report, and then 2 behavioral reports, again, number of thoughts and percent of correct count, and then alpha band power, and then auditory evoked potential amplitude. Yeah. Based on a percent mind wandering, we actually define high mind wandering report and then low mind wandering, just a focused report. And then from this point, we actually cut the segment and then align them and then average. So we get two average signals, average data, which one is a mind wandering plus minus 30 minutes from a mind wandering report, and then the other is task focus segments. Again, the plus minus 30 minutes from the focused report. Sorry, 30 minutes. This time is not 8 hours. It's because it's an easy experiment. We cannot measure, keep that long. Okay, so those data is fit to the model, which I will explain in the next slide. Then for this, through this, we actually estimate the direction and timing of neural effects for mind wandering condition and focus condition. Right. So the model is very simple. This is basically one model is a psychological response of number of thoughts is explained by two neural predictors, alpha amplitude and then AEP amplitude, and then one sort of participant. This is a random variable. And then from this model, we actually have two reduced models. For example, this one comparison of 1 and then 1.1 gives the contribution of alpha activity to a number of thoughts. Okay. The another thing is we also have this time lag, this L, to evaluate the timing of the effect. So we changed the we sort of shifted the segments to the past. Yeah, sort of to the past in the 2 minute cycles, so 0 lag to 2 minutes before, and in the 4 minutes before, 6 minutes before, and so forth. So in this way that we actually can assess the what timing, what kind of time lag is effective for this to explain this psychological response. The same thing for the percent correct. So I say, okay, so let's be more concrete example. So models, if I compare these models 1 versus 1.1, this gives the effect of alpha band activity to the level of to the number of thoughts. And then, so in this case, lags were changed from 0 to minus 30 minutes. So, which this gives this type of representation. So if the effect, positive effect, neural effects, positive effect or negative effect, and then how this is a simultaneous, and this is a neural effect, neural activity of 2 minutes before is indeed affecting them in negative manner, negative, positive, and so forth. But if we actually apply some sort of statistical control, in this case, this one is the only, say, reportable, somewhat reasonable effect. So this, in this case, 18 minutes before the actual report of mind wandering. Okay, so we do this. So for all these stuff, then we get this type of results. So this is a mind wandering condition, and this is a task focused condition. This is a bit confusing, but this is the estimated time lag in the maybe I just one back. So okay, time lag. So in case of the 0 to 20, so you can probably have more multiple peaks here. No? Okay, so what do I have? So these are the time lags. Yes, indeed, there are quite a long time quite a lot of neural effects which have a quite long time lags. And this is task focused condition. So, and also they notice this with the positive effect, but this one is changed to negative effect here. This is positive. So, yeah, in fact, these neural mode effect, timing, and the direction of those neural activities are not constant and not always antagonizing. So from this, we can say we can add a few more things to the slide. Yes, mind wandering is unfolding in space in real time as a trajectory, and the trajectory is affected, modulated at least in part by the alpha band activity and the event-related activity of the brain. And the time lag is somewhat longer than what we are used to. It's not 300 milliseconds. It actually is in the time scale of minutes. And then the most important is the timing and the directions of the neural effects are variable, at least in the domain of spontaneous thought process. Okay. Right. Okay. So neural network. I actually added this slide, but I have a bit of a reservation because this is a very early stage of results, which actually is one student's master's thesis. And we are not 100% sure, but it may be interesting to present it. This is slightly different from a previous analysis. But what we did was we used the same data of the tone counting task data, and then we applied the independent component analysis, and then we sort of classified those ICs to either evoked components or non-evoked components. Having this, we can sort of think of the networks amongst the evoked ICs, this yellow one, or amongst non-evoked ICs, red one, or between them, right? And some studies were reporting that the evoked ICs increases its say synchrony among them, so sort of not increasing, decreasing it with the number percentage of mind wandering. But some of the papers, at least Jacobs and I read together, had many methodological problems. So we actually tried to do it so more, how can I say, proper manner. And the pairwise synchrony was measured by a single value PLV, and then easy band, alpha, theta, and gamma. We took all of them. Then we are also sort of expected the evoked IC will decrease its synchrony, but so kind of non-evoked ICs might increase the synchrony with the percentage of mind wandering, but no, that was not the case. So far we didn't find any of the, we didn't replicate the studies which were reported earlier. So that actually is the first part. And then you might think, what actually, but yeah, maybe this. But the interesting thing maybe is of this, the highest synchrony is among these non-evoked ICs. Yeah.
And Chie?
Yeah.
We are basically on time.
Okay.
If you have something more, then we can.
Oh, okay, sorry. Yeah, no problem. That's okay. Then yeah, this is not really important alpha. Yes, this is basically the same scenario, same stuff. Okay, so dynamics. Okay, what we actually can say is the mind wandering is a unfolding in 9-dimensional space in real time as a trajectory, and the trajectory is modulated by neural activities, but the time lags are probably longer than we think of, and then so those things are variable. So that is, yeah. Thank you.
Thank you, Chie. It's incredible how many things our minds do while we think we are doing nothing. Okay, so now I'm going to introduce the next speaker. Thank you, Chie, again. Devia is an academic researcher at the Department of Neuroscience of the University of Chile in Santiago de Chile. She studies active sensing, neural activity associated with free visual exploration of scenes, and perceptual processing during the exploration of natural, artificial, and/or face images. She is especially interested in the role of neural coordination as a mechanism of perception and the ensuing activity locked to the eye movements in health and psychiatric diseases, and brain dynamics and consciousness through deep anesthesia in adults and children. Her talk will be about neural correlates of natural scene perception. Chris, when you want.
Hi, Chiara. Thanks. So my original title was Neural Correlates of Natural Scene Perception, but I changed it to Behavioral and Neural Correlates. You'll see why. So let's start. Oops, sorry. I'm sorry. This is at the last. Sorry. Don't look. There. Now we are at the beginning. So let me start telling you why I'm interested, or I think that natural scenes are a good paradigm to study visual perception and brain processes. So when you look at this image, you usually will do like 3 or 4 movements of your eyes on each second. And if I don't give you any instruction, you won't be able to control this. So we all humans and primates and other animals, we move our eyes all the time. So usually you will do something that we call fixation. That's when you spend some time looking at a specific area of your image, and then you will move here. Do you see my pointer? No, now you see my pointer. So you will look at a specific area of the image, and then you will do a saccade, which is a fast movement to another position, and then you spend some time there. So, and this keeps going all the time. But we have seen that this sequence or pattern of movement can be affected in some patients. In this case, what I'm showing you on this column is the eye behavior of control, healthy controls. And here I'm showing you the eye behavior of patients. In this case, in this specific case, these are schizophrenia patients. And you can easily see that, especially in natural scenes, controls move the eyes around the image, while patients tend to keep their eyes in a small area. So we first went there and looked at the number of saccades that patients, here in green, and controls perform over images, and we realized that they don't have significant differences, not for natural scenes, not for pink noises, and not for black plain image. But when you look at the total distance covered by the movement of the eye, you can see that there is a strong differences, that there are strong differences between patients and controls, particularly in the case when they are exploring natural scenes. Okay, so there is something about this kind of image that somehow let us see differences in these two populations. So then we went further and we studied the EEG activity. So here I'm just showing you part of the results. But we were able to see that over occipital electrodes, particularly this one, this area, the posterior area of the scalp, we see that controls have an incremented ERP around 400 and 600 milliseconds, which is not apparent in patients. So you can see here that in the scalp topography and also in the ERP, healthy controls have an increment in the ERP that is not present on the patients. So again, what they are looking at is at a natural scene. So at least to me, this is really like important. There is something about the free exploration of natural scenes that could even be a tool in the future. I mean, it could even be a tool to support psychiatric diagnosis. So that's a motivation enough, at least to me. So, but what happens or what you do with your eyes when I show you a natural scene? Well, of course, you move your eyes, but there is a pattern. So what I'm showing you here is the number of saccades. So that's the number of movements that you perform over the image. And this is the time from image onset. So at time 0, image appeared on the screen. Before that, there was a gray screen, plain gray screen. And at 0 appeared the natural scene. And you can see that we have like a stable number of saccades on the gray image, on the gray screen. So it's like a stable number of saccades, around 100. Then appears the image, and 100 milliseconds after image onset, you see here a huge decrement in the number of saccades that you perform. That means that if you're not performing a saccade, you are in a fixation. That means that you put your eyes in a steady way, in a steady but particularly and systematically across all the images. Around 100 milliseconds, you keep your eyes fixated. And then you start to move again, and you move them a lot more than you used to move them in the previous plain screen. So that was really interesting to us because similar results have been found for monkey data. Here, what I'm showing you is time from image onset, but the authors are showing the number of fixations, which is complementary to the number of saccades, as you might guess. So you can see here that you see the same. So there is a decrement in the number of fixations that you perform, and then the onset. So what we can get from these images is that after 100 milliseconds, you change your behavior in response to the image that appeared on the screen, okay? So then thinking about this like really stereotype movement of your eyes, we said, okay, maybe we can predict the scan path. So where are you going to move your eyes on the image while you're looking at a natural scene? And so Camilo started to work in several neural networks models. I won't go into the details. What I'm just going to tell you that here I'm showing you in blue. So let's see this one. In blue is the original eye movement of the subject, the scan path over the image. And the other colors show different models tested, trained with natural scenes, and then tested with other natural scenes to predict the movement of the eye of this subject. And as you can see here, it's just trying to predict one step ahead. So that would mean like around 2 milliseconds, okay? And it's reasonably good, but here is trying to predict 5 steps ahead, meaning this is trying to predict the next 10 milliseconds of movement, which is a lot for this kind of data. So as you can see here, there are some differences in the position that the models predict, but at least it's able to predict in general the scan path. So we quantify the differences. I will just show you the details for 2 models. I won't give you all the details of the models, but one is recursive and the other is not recursive. But the main thing is that if you concentrate on these 2 graphs, I'm showing you here the error in the y-axis, so in the prediction in the vertical component of the movement. And you can see that when it's trying to predict 1 step ahead, it usually it makes an error of 10 pixels or less. So you can see here that almost all the tests done had the density below 10 pixels. And also in this case. So when we move forward and we are more ambitious and we try to make predictions 5 steps ahead, you can see here that we keep this error. So the models are quite good predicting the position of the eye movement. Yes, this is within this error is around 1.25 visual degrees. So it's just like really small error. So after saying that, I can show you the general performance. So please here, look at my graph. I'm showing you here when we are trying to predict 1 step ahead. What I'm showing you, this axis just shows you how much is the error. So of course, less error is better than more error, okay? And what I'm showing you on this axis is the images that we use to train. So when it says natural, it means that we took all the behavioral eye movements of the subjects when they were looking at natural scenes, and then we tried to predict the others. Yes, so the models were trained with this kind of data from this kind of image. And you can see what I would like you to get from this is that pink noise, white noise, which is another kind of noise, inverted image, so images upside down, and natural scenes, all of them are much better, have a better performance than when you use plain images like white, just black, or just gray image. So again, there is something about the natural scenes that is better, and the model is able to capture that on the training. But this also shows us that it's not just natural scenes, it's any scene that has texture. So any scene, any image that has high frequency or higher frequency power when you evaluate the image. And we observe the same results when we work with the 5 step ahead models. So again, texture images are much better to train a model than plain image. So just like what I show you for the patients, we have something on natural and texture image that it's really different from the plain scenes. So, but what are the neural correlates of natural scene perception? What I'll show you next is the really simple task. Subjects look at a screen with a fixation cross, and then it appears an image. So the image could be a natural scene, like this one, and inverted, so you can see that this one is inverted. It's not that clear, but it's inverted here. This is a pink noise version of the image. This is a white noise version of the image, and they will also see white, plain gray, and plain black, okay? So the task was, please explore the images. We might ask you something about the images at the end. So that instruction was just to keep them engaged on the task. So what do we see? For natural scenes, so the first one that I show you, we observe that when image appears, so at this scalp map, when image appear, we observe an increment around 100 milliseconds, but then a huge increment in occipital electrodes on posterior in general electrodes at 200 milliseconds, okay? When you compare this, so you can see here clearly this huge lightning. When you compare these scalp maps with white and black images, you see that after image onset, so here this one is around 0, this one is 100 milliseconds, so all of them around 100 milliseconds show some increment in the evoke activity, but not that much. But then at 200 milliseconds, only natural scenes have this huge increment in activity. Yes, you don't see that for plain images. And this localized increment is sustained in time, okay? So just remember that the subjects can explore the image, so they have the image in front of them all this time, okay? Finally, if we look at pink noise, so which is an image that doesn't have the semantic content of natural scenes, but does have the same power content in the image, physical content, we observe similar results that for natural scenes. So we have this increment very localized at 100 milliseconds, and then this huge posterior increment in evoke activity at 200 milliseconds. So you see that this one is quite similar to this one. When we look at the ERP over the electrodes, we see we can check the same result. So you see here the 100 millisecond increment. So I'm showing you here time. This is voltage. And in blue, you can see the occipital electrode. You have all the others, some others in the midline here, but we'll keep our attention on the occipital one. You see the 100 peak and then the 200 peak, okay? And if we compare with the white image onset, so here is the gray screen, and here appears a natural scenes after this line, and here it appears a white image. You can see here also this increment, which is almost equivalent to this one, but then you don't see this next modulation, just like a sustained activity. So far what we have seen is that there is like a first response around 100 milliseconds for natural scenes, and then a huge response at 200 milliseconds. But what happens in frequency content? So now I'm just showing you the OC, the occipital electrode in the midline. Here I'm showing you time. So 0 is image onset. Here I'm showing you frequencies. And the color scale shows you the power on decibels, but it's normalized from the baseline. So this is baseline. This is again the gray screen with the fixation cross. And as you can see here, what we observe is that after image onset, so let's look at this one first, which is the natural scenes. We observe that after image onset, we have an increment in power, which is brief, fast, and then a decrement in power in this band. So this one like appears and then goes down very quickly. Complementary, we observe this other, like below alpha or at 10 hertz, below 10 hertz, we see that we have an increment in power that is sustained. So it decreases, yes, but it's sustained in time. This one, we don't see it here in the white image. So natural scenes have different frequency content in the occipital electrodes. So I want you to focus on this, because this part, like from what we can see now, what I'm showing you now, this part is similar. So white images, natural scenes, and we have like a similar dynamic. And this is between 10 Hertz and 20 Hertz. So again, we have this brief increment in power and then a decrease. And the different part is this one. Okay? But to test this, like make an statistical test, we compare this to the power, I mean the spectrogram of the two categories using a t-test comparison. So this is not actually a t-test, this is a cluster mass permutation test, but the main idea is that you get the cluster, you make a simple t-test, pixel by pixel, and you get the clusters that are significantly different between these two spectrograms, okay? So that's what I'm showing you here. So let's going to look with details this comparison. So what we have here is, again, this is time, this is 0, and these are frequencies. So this is the baseline. Again, this is the gray screen with the fixation cross. And at 0 time, appear the image. And what we are looking here is the, on this scale bar, is the T values. So it's the difference between natural scenes and white images. So a red color would mean that at that moment, natural scenes have more power than white scenes. And a blue color would mean the other one, the other way. And as you can see, this is 100 milliseconds, and you see here that the test doesn't see any significant statistical difference between the power for natural scenes or white scenes, okay, or white images, sorry. So you can see here that the test doesn't see any differences, and all the difference that the test found were after 100 milliseconds. Okay, and if we have this like really huge activity that is bigger for natural scenes than for white images, which is like up to 15 Hertz, I think, like alpha band. And then we see in beta band that there are two significant clusters that are showing us that white images has even a stronger reduction in beta band than natural scenes. Okay, so so we move forward because here we are comparing natural scenes with white images. But I told you from the work of Camilo that at least the neural networks that we are training to predict the scan path, they can predict equally good from all the texture images. So we said, okay, let's look at the other texture images, compare with the white one. So that's what I'm showing you here. So this is again the same comparison that I showed you before, natural scenes compared with white images. But what I'm showing you here is pink noise compared to white images and white noise compared to white images. And you can see again, so this is 0 time, the scales are all the same. This is frequency, this is time and frequency. And you can see here that we see the same general pattern. So for all these texture images, we observe that after 100 milliseconds, right after, we observe an increment in low frequencies below 10 or 15 Hertz, and a reduction at beta band, but that this reduction is always bigger for white images. So to me, this was really interesting because you can see that this is a similar pattern here. So all the texture image images show a similar pattern of power like equilibrium or balance. But then we have something else, and if you see this dashed line that I put here is 30 Hz, which is usually like the limit for beta band and to start to talk about gamma band. So you can see that for natural scenes, we don't have any of that. So everything is below 30 Hz. But for pink noise and for white noise, it's not the case. So they move like upper a little, upper on frequency. Okay, we don't have like a hypothesis of this result yet, but it just seemed interesting to share it with you. So what we have seen from this last part was that we have like a brief increment in low beta power band. So this is around 100 milliseconds when the natural scene appear and when the white image appear. But then after 100 milliseconds, that's the moment where we see the differences between the processing of a plain image or a texture image. And we see that this dynamic processing of the image after 100 milliseconds is heavily supported by delta, theta, alpha, and part of beta band. So in general, just to finish my talk, the final conclusion of all the work that I have shown you is that we have like here, like this timeline that I'm showing you here. We have 0, which is the time of image onset. Here in the upper part, I'm showing you the behavior dynamics, and in the lower part, I'm showing you the brain dynamics. And you can see that on the behavior, I show you that between 100 and 200 milliseconds, there's a reduction in saccade rate, and there is a huge increase in the saccade rate after 200 milliseconds. On the brain dynamics, we see that between 0 and 100 milliseconds, there is an increment in power, and that this increment is between 10 and 20 Hertz, but it appears on plain images and also on natural scenes images. Then after 100 milliseconds, we observe this sustained low power increment and a sustained beta power reduction. And why do I emphasize this? Because we have this like interpretation that we are trying to give the strong evidence about, that this first 100 milliseconds, you are like in a feed-forward mode. So the image appears, you don't change your behavior, you don't have time to do that, you don't have time to do anything, you're just getting like the gist of the scene. You're in a state that something appeared to you and you have to like make a first, let's call it a decision. And then it starts this, then you change your behavior in response to this image, and then you change your neural dynamics in response to this image. So with that, I think I'm ready. So I would like to give thanks to all the supporting institutions and to all the people that work with me for this particular results, experiment, analysis that I showed you here. And that's it, I think. Thank you.
Thank you, Chris, for your interesting presentation. I really enjoyed it. As all the presentations in this symposium, I'm really happy. So finally, we are coming to the last speaker. In Latin, it will be Dulcis in Fundo. Pedro Maldonado is professor and researcher in the Department of Neuroscience and part of the Biomedical Neuroscience Institute, BNI, at the University of Chile in Santiago de Chile. His main topics are active perception, visual behavior of neuropsychiatric patients, and the homeostatic energy principle as configuring neural networks functioning. I am looking forward to listening to his talk about a speculation on a biological and evolutionary role of consciousness. Pedro, the stage is yours.
First, I want to thank Chiara for organizing and putting together this symposium. It's been a while since we had this quality and in this setting, so I thank you. I would like to share with you some speculation, and I would like to stress speculation because we would like to let you know about some reflections that Rubén Moreno Bote and I have been thinking about consciousness. And consciousness is a very interesting topic, but of course, let's start with the obvious. But as scientists, we need to define the phenomena that we need to explain. And if you look around, consciousness is something that does not generate a lot of consensus. So everybody will have their own idea what consciousness is. So if we look around for definitions from Wikipedia, for instance, the idea is that this relates to knowledge, and typically you will find the idea that consciousness relates to the knowledge that you can take or you have that a being has himself of his surrounding. This doesn't really help because we move from the concept of consciousness to knowledge, which is equally ambiguous. But in general, you will find definitions that relate to some awareness, right, of an external object. So it seems to refer somehow here to some subjective experience, the ability of experience, feel, awakeness, pain, vision, but also the experience of feeling some sense of identity and so on. Now, in neuroscience, the common expression of consciousness is the physiological state of wakefulness, and Srivas here gave us a great talk about that. In psychology, this is more about behavior and how humans relates to himself and the world. It's important that we make a distinction. In English, there's two words that have been often used here, which is consciousness and awareness, and we don't have a specific word for awareness in Spanish, so darse cuenta may be the closest thing to that. In psychiatry, consciousness is also used as a cognitive states, but here are used in a context will they stress the interaction and interpretation of external stimuli called reality, right? And in that sense, for psychiatry, the consciousness require the use of our senses to connect to the world, and this is important as you will see. So if you overall look at all the definitions, the common denominator basically relate to the fact that consciousness is in general defined as a subjective experience. And Andres mentioned this in his initial talk, right? And I completely agree with. But subjective experience are nothing new for us. We have a lot of subjective experience. We feel pain, we see, we hear, we experience emotions, and so on. So in this sense, I will start from the fact that I will consider consciousness as one of many other subject experience that we as organisms have. And so far, there doesn't seem to be anything special between when we compare consciousness with other subjective experience. They are basically the same basic phenomena. A phenomena that, of course, we all seems to agree that is caused by the brain. Without brain, we don't have any consciousness. So can we explain consciousness? Well, can we explain subjective experience? Yeah, of course. I mean, we can try. We're trying to explain some of our subjective experience. but not everybody agreed. There's people that, even says that this is just a philosophical concept, or it's very complex. It's not, we cannot really understand it, or we're not even capable of doing it, or yes. And in the study of consciousness, there's 3 types of approximation, I would say. Wakefulness versus unconsciousness, the idea of the qualia, and the hard problem, okay? So I will go through this. Srivas show us a wonderful talk about how consciousness can be explored from these very basic 2 states, right, of wakefulness versus unconscious. And he showed experiments in which you can examine neural activity to distinguish between these 2 and understand the neural correlates of the dynamics that move us from one state to another. But this is not what I go on after. And here's an example that he showed a few ones, but here are different metrics that you can use and how this can be then or used to show that as you move from different states of consciousness, the brain will function in a different way. And a second aspect of the consciousness is the idea of qualia. And qualia refers to the ability of humans to communicate about their subjective experience, right? And this is the wonderful things about human, right, that can share the experience through language. And this, of course, enables experimental access to neuroscience. So somebody can ask me, what do you feel? How do you see? And what is your conscious experience, and so on. So in this way, there's been a lot of study of consciousness in which we can study the neural correlates of the reported experience, whether we see colors or an image or a sound or feel in a particular way or etc. So one of the questions we would like to understand is that, well, since this is cause of the brain and generate a objective experience, is there any particular part of the brain that generate this particular subjective experience? I mean, we know that the visual cortex may generate visual experience, and the olfactory system will generate all others and so on. But what about the consciousness? Well, we haven't found particularly any part of the brain which seems to be the unique correlate of consciousness. And in that regard, this is interesting because it will say that many different brains could generate effectively this subjective experience. Why humans that don't have any particular different structure from other animals will be the only ones that experience subjective experiences? We know that other animals also exhibit an subjective experience like seeing and pain and so on. So there's no reason to exclude other animals for the brain generating the subjective experience that we call consciousness. And in fact, many years ago, I mean, you probably know about this Cambridge Declaration where this group of scientists make the statements that there was no reason to think that every other animal may have consciousness, right? And while it's not obvious to the world, it is obvious to society. And finally, I would like to mention the idea of the hard problem, right? That is an idea put forward by David Chalmers, and he defined exactly the difficulty of the problem of consciousness is the problem of explaining the relationship between physical phenomena such as brain processes and experience. Now, why are physical processes always accompanied by experience? And why does a given physical process generate a specific experience it produces? And why an experience of red instead of green? And if you think about this, I would like to propose that the hard problem is not a problem private to consciousness. In fact, every subjective experience suffers for exactly the same hard problem, we feel like what is seeing, but we cannot explain how the activity of the visual system resulted in this subjective experience. We know that some part of the brain light up when we feel pain, but the experience of pain is something we cannot explain only by action potentials and neurotransmitter. So the heart problem is not privative of consciousness. All subjective experience have a heart problem. And we are very, very far away to understand how pain or a visual experience or an emotion results, it raises up as a consequence of neural activity. Consciousness is just one more of many different types of subjective experience. So then this will bring us to the reflection that, okay, if visual experience, subjective visual experience is the activity of a neural system and pain and so on, the question is that what is the neural correlate or the neural activity that will give rise to subject experience? So we have been discussing with Rubén Moreno Bote, which I have here a picture of, and we have come with a proposal, with a speculation that we'll present to you. And this proposal, this mind-wandering, start with the idea of decisions, right? We all make decisions all the day, right? All along. And a decision is typically a conclusion, a resolution that we reach after a consideration. But what is a consideration in our brain? We know that when we make decisions, basically many of the neural systems are active, right? From the cingulate, the nuclear accumbens, the prefrontal cortex, of course, but also sensory cortex depending on the type of decision we need to make, and of course, amygdala, hippocampus, and so on. So apparently making decisions requires large portions of the neural networks involved in different type of behavior. So why? Why it will require this? Why we don't making the decision in a small area of the brain? There are many models about this, the typical model of data accumulation and information accumulation between binary decisions, but I'm not going to discuss these different type of models. But what I like to stress is that to make decisions, it is necessary to evaluate different scenarios presented by the alternative to be decided. Basically, what we observe as organisms become more complex is that the pool of different behavior that animals can engage in are more larger and larger. A very simple organism does have a very small repertoire of behavior. So it doesn't have to choose between different types of behavior. But as our brain becomes more complex, there is a huge number of different potential behaviors that we can engage on. So we need a mechanism to decide which of these behaviors we're going to do. And to make these necessary decisions, we need to evaluate the potential consequences of each of these decisions or course of actions in behavior. And to make the right choice, we need to evaluate what are the consequences of taking behavior A or behavior B. So it seems necessary that our brain engage in a process of emulating different scenarios. But these processes requires brain resources. Do we have a different brain resources to make an evaluation of a particular decisions? So we need to evaluate about which food to eat. Do we need or we use some additional resources, or we use the same visual system, the olfactory system, the emotional system to evaluate this alternative? I mean, this last thing seems to be the reasonable thing to do, right? We have only one brain, which is very expensive. So if we have a huge network, it seems that only one that we use to generate all our behavior, it will make sense that we use exactly the same network to simulate in our brain the consequences of future behaviors to make proper decisions. And in fact, many studies have shown that imagery or rehearsal or memory or evaluating decision seem to employ exactly the same networks that we use during the actual behavioral act. So for example, in this experiment, when we realize a particular movement and then we imagine the same movement, these authors show that the same network basically were active in a very similar way. The same way when you encode things in memory and then go through the process of retrieval, studies show that you tend to then recruit basically the same areas. If I ask you to remember a visual memory, it is likely that most of your visual system will engage in this memory process, recreating the elements necessary to fall in this particular visual state. So if decision-making requires simulations performed by the brain, more complex decisions require more complex simulations. And interesting, the bigger your brain and the more complex your behavior, the more complex your simulation. Our brain is able to establish or recreate or simulate very complex situations, from what we're going to do next year, what would happen after we have lunch, how we plan to meet with somebody, what we're going to say, etc. So notice that our brain in particular have at their disposal a huge network to simulate very complex situations, and experience these simulations in a way that is very that resemble our real interactions with the world. If we plan for a meeting for getting a new job and examining what we're going to say and the emotions we're going to feel, we actually really engage in that emotions and so on. So simulating and experience the world basically are two situations in which we use the same brain networks. And so we can either simulate or interact with the real world. And when we simulate, we seem to disengage from the world. Think about the many times we need to make a decision. We close the eye, we move the head, we look to the ceiling. We try to disengage from our real interactions to maybe employ our networks to properly engage in these internal simulations. Right, but now we have a problem. We require the brain to perform internal simulations to make decisions or decide which behavior we're going to engage in. But at the same time, we need to be aware of the environment and what things are going on. So if our simulations are very good, how we distinguish a simulation from the environment with the real coupling, right? So what we propose here, and I'm going to share this, is that basically there is a neural mechanism that monitors coupling with the environment and monitors when we are engaging in a simulation and when we are coupled with external world. If we don't have this mechanism that is actually to monitor, we cannot distinguish between brain simulation and the real world. And this actually do happen in psychiatry a lot, right? We know many cases of this. So the brain must have a mechanism that is basically able to monitor and distinguish these two. The activity of this brain mechanism leads to a subjective experience, and we propose to call this subjective experience consciousness. So this is not the first group that actually are proposing this. They're like Hock and Law, the UCLA, that also hint at this a little bit. But in our view, this concept has some corollary. One corollary is that in dreams, we have no consciousness. And in fact, at least this happened to me. In the dreams, things happen, but I don't have any conscious experience of, no, this is not real, though. I don't have a reflection in this They just happen. And of course, you don't need to distinguish between reality and simulations because you're dreaming, right? You're in a state that this is not necessary, right? Of course, because this is a necessity to distinguish between reality and simulations to make decisions about behavior, all animals may have consciousness, but the expectation is that the less complex the brain, the less complex the simulation, and probably the less complex the experience of consciousness. In this regard, consciousness may be a gradual experience, not you are conscious, you are not. You may be a little bit conscious or not, depending on the balance of your engagement with the external world and your internal simulations. And of course, the experience, the subjective experience, should happen after the particular neural correlate engaged in this monitoring. It's not previous to. With that, and I will actually leave with that because this is the take-home message that we want to convey. I hope that you have enjoyed this idea, and we'll see what consequences this should lead in the future. So with that, I took less time, but I know that people are tired and probably want to go in discussion or just have lunch. So with that, I would like to finish my presentation and of course, thank to many people that have contributed to the science that lead us to these ideas. And I thank you for your attention.
Thank you a lot, Pedro. It was really interesting to listen to your proposal. I have many questions myself. So let's see if we have actually many questions for the second round, the second session of talks. We have published them. So here, yeah, the first one is for Chie Nakatani. The question is, according to your understanding of the mind-wandering functioning, when would you consider more probable to experience déjà vu events, the phenomenon of déjà vu, during mind-wandering or while being concentrated in certain specific thoughts or performing a task? Thank you.
Oh, yeah, okay, yeah. It's an interesting question. I never thought of it. And déjà vu is, I do not even know how it is in fact defined in the space. And it involves certainly some sort of memory component. And it actually is not generating something like, but more to, yeah, so yeah, the short answer is I don't know. Seriously, I really do not really know the if anyone in mind wandering has any déjà vu experience. But okay, so the in my very limited data set, which is a answer from college student in Belgium, and they are very nice people, and then they're active, and what they are mind wandering about, that's somewhat similar to what other speaker said. Okay, it is not very far future. It's actually a little bit in future, and then something quite plausible. So it's like a simulation, like we just heard from the last talk. So those are the things which is occupying their mind when they are mind wandering. So it's not like a déjà vu type of thing. It could occur, but you know, it was relatively, or I didn't see that type of report in my data so far. Did I answer to the question?
Yes, thank you. But if someone else of the speakers has an idea or want to say something, of course, you can also answer this question. If someone, no. So there is another question that I think it's for Chris, Devia. Thank you for the presentation. A naive question. Is it plausible to connect this lack of exploring a natural scene in schizophrenic subjects to their tendency to assign affective balance and meaning to neutral percepts? For example, positive symptoms linked to specific elements in the natural scene, rather than broad contemplation of the scene?
That's a hard question. And a good one. I'm not sure. I'm not sure. I would say that possibly, but we don't have any evidence, like clear, strong evidence in that line. But I would say that we've been thinking that in a future, not now, but in a future, you might be able to develop some kind like a behavioral therapy that you could teach to these populations to look at the scene more broadly, and maybe that might help, but we don't know. We are far away from that.
That would be interesting to see if it's possible. Thank you, Chris. So there is another last question. I have a question for Chie Nakatani again. Mind wandering is often associated with default networks activities in brain. Could activity in default network be related to clinical conditions like attentional deficit disorder or depressive disorder?
That's the million-dollar question everyone wants to know. Is that there are two problems. First of all, the default mode network is defined in a different way, and then people use it in a slightly different way, and then in a way that they use is very relatively conceptual. So, and also it is not something network. So you say you do some visual task and then some resources or brain structure activities are used for something, but which is not used or deactivated. So it was a default mode network. If you think about it, it's very difficult to sort of grasp the gist of what default mode network does. So that's the number one problem, conceptual problem, but then we have other interesting symptoms and so forth, and then they also have quite the multiple. The word nebulous is not so great, but it's in a way amorphous type of definitions. Okay, so so how to how can I say link those two ideas together in a way that we can study is in a way very big question. So we need to somewhat start from that. That's what I realized when I started to do this mind wandering study. So then that somewhat motivated me to come up with this psychological functions, very simple ones, and then very simple understanding of what brain network could do and how to, and then let them work, then how they can sort of, we can compare them. That's a kind of approach that I am now after. Because that is because it's a difficulty of, it is difficult. I came to this approach because other approaches which I'm used to study attentions or how can I say fixation sequence and so forth didn't work.
Yeah, thank you for the answer. Also, again, if someone has something to say about this, of course, you can. Now it's our turn. If someone of us has something to ask to the others, we can also make questions.
I have a question for Pedro, if that's possible.
Of course.
Yeah. So, do you have any kind of favorite or potential mechanism by which the brain is actually doing the monitoring contrast? Because in your last statement, I saw like there is like the underlying assumption that there is such a mechanism, so it exists and it's implemented in some way in the brain. So, do you have any potential candidate for that, or is it just too early stages for that?
Thank you, Andres, for your question. I mean, you got me. This is one of the things that it's actually very difficult to assess. We know that in any behavior or cognition state, we can identify typically which part of the brain may be involved, but we have not really clue on this one. Because there doesn't seem to be a particular area of the brain that do the monitoring. However, there's some hints that prefrontal cortex that relates to executive function may be also used in a similar way. We know very little about prefrontal cortex. I mean, there's many different parts, but we cannot disentangle the contribution of each different part. And I think that I suspect that this may be. One of the problems we have is that this monitoring system will not work unless you have the ability to make the simulation or be alert. So if you kill the thalamus, of course you have you cannot see this because you are not conscious anymore. So as many other cognitive functions, we know that they're depending on large networks. And so if we knock out one piece of the brain, we typically have consequences that are not easy to separate from other functions. So then this is a problem. So I don't know. I mean, this is just hand waving at this point because of course I have no idea. But it will be interesting to maybe think more to propose a particular experiments in which this monitoring should be specifically identified, and then see what part of the brain are critical, at least for this to happen.
Mm-hmm. Thank you. So at least you consider the frontal cortex as a potential important part. And I think that's different from main theories of consciousness, that there are some theories that claim that the relevant part of the brain is the back, not the front.
Right, but of course what happens is that when you're visually conscious, you need to light up the occipital cortex as well, you know. But one interesting thing is that the leading theories, actually the ITT and the global spacework theory, they don't make the relationship to the subjective experience, and that's the thing. But I think it's really interesting that we have at least two ways of use our brain: to interact with the world, but also to mind wandering, but mind wandering in a necessary way, because our brain, when it's not engaging in direct interaction with the environment, have the ability to still using it to probe and to explore and to understand, and it will make sense that these abilities are really as complex as the way we interact with the environment. And if we cannot tell the difference, and this I may relate to the questions that somebody made to Chris. In schizophrenia, one of the hallmarks is the ability to lose reality monitoring. So apparently consciousness it may be very critical to be studied in this group of patients. I mean, reality and imagination for some patients are undistinguishable. And why is their system not working? You know, apparently this relates to one possibility is that a deficiency in the long-range connections in the brain that makes the brain probably function together. So there's this hypothesis of aberrant connectivity, which is gaining terrain in schizophrenia, which basically means that you cannot, the brain of these patients are not able to work together in the proper way, whatever that is translated. Anyway, I will end here.
Thanks, Pedro.
Someone else has other questions to the other speakers? Otherwise, I will ask my question to Pedro. I really was interested in your proposal because the monitoring system would overcome some problems. I mean, when you think about consciousness or when you think about the proposal, then you always have a question that is, okay, if the processing of sensorial information or coming from the outer world gives energy, takes energy from outside and is processed in the brain, so the energy comes from outside. Then when you are the other way around, where does the energy come from? So if the energy is self-produced, I mean, is recurrent in the same system, then of course it solves at least this problem that for me was a kind of problem, philosophical problem. So if you imagine that this system would, simulation system would automatically start. So whenever I have a task from the outer world, then the simulation is automatic. But I can also decide to consciously simulate something. So it's when I plan my workshop or whatever. This is complicated to me because it's automatic, but also I can top down. So there, exactly, when I top down decide something, or I'm voluntarily changing what I'm seeing in case of bistable phenomena, for example, or whatever, where does the energy to do this change or to send to the monitoring network come from? So still I have this this kind of problem, and I was trying to relate the default mode network, of course, because we know from literature that exploring the default mode network activity in autism or in schizophrenic patients, of course, we see differences. So we are close to that. I mean, they are similar, or at least they work together. But this part, when I voluntarily change this, what happens there, according to you?
Yeah, what a wonderful comment. This is very interesting what you mentioned, and well, if you look at the brain of organism complex like us, you wonder the question why you need more brain than a monkey, you know, and basically the function of seeing and touching and hearing do not require a brain as large as ours. So that means that our brain is very powerful, and of course the primary function of the brain is to keep us alive, so we need to engage to the environment. But to process things in the environment does not take as much brain as we have, so it seems that our brain was enabled to do more than just engage with the environment. So biologically, it will make sense that if you have this powerful machine, and then your brain decided that there's no immediate threats or need to interact with the environment, you could use this brain for something else, like exploring internally of wandering or simulation of or what we go into the default mode network and so on. Now, the question is who decided when we do one thing or the other? Probably the neural mechanism that is responsible for consciousness is probably the same mechanism that makes this decision, because you cannot freely engage in mind wandering or in simulations if something else is going on outside. You will not live longer, right? Because you will not be able to see the threats. So I imagine that the monitoring and the decision making to in which state we are likely to be related anatomically and physiologically. But I think it's a very important mechanism that is not only a monitoring system, but also you have a system to switch or make decisions in switching between these two modes. Which I have no idea about how this may work, but I think it's a very important point you make that these two things should be very closely related.
Thank you a lot for your answer. And also I have another, let's say, specific question. When you alter your consciousness with well, we say drugs, but including psychopharmacs or pharmacological things. So you are obliging your networks to work or work differently. Then you are able to do things that usually you don't do. So, for example, I've heard some colleagues that say when you take these mushrooms, peyote, I don't know how it's called, then he was able to distinguish and completely perceive separately all the flavors of the sandwich that he made, and he made really an awkward sandwich, like with marmalade and salami and cheese. He put everything, and he was not disgusted. First of all, this is very strange, but he could chemically distinguish everything, every component of the sandwich, and but he was on peyote. So my question goes through: do you think that there are some barriers that our own brain puts on ourselves, maybe the frontal cortex, that if we remove these barriers, then we could do something more that maybe we didn't explore, for example, with consciousness?
Well, I really like your questions because one, I like it because the exploration of the brain on drugs can give us a window about how this may work. Now, this is not an easy question because people that are on drugs, marijuana, peyote, they later relate that they were completely aware. But this is a tale that they do completely after these things wear off. So people in marijuana says, oh, I completely lose it, I see. But when you test them, they don't perform any better. I doubt that the people can distinguish the in single odorants of a sandwich because our olfactory system does not have this type of ability, honestly. As wine connoisseurs say, you know, I distinguish this and that. So the question I would like to ask these people is, what type of consciousness do you have while this? Because were they conscious in the same that we are conscious, that we distinguish what is real and not real? Are they able to make this use of this monitoring system? Maybe the use of drugs alters many things, among them the ability to understand when you are engaged with the environment, when you are in internal mode. So the problem is that all we know is what they tell us after they are off the drugs. So it's technically difficult but it will be great experiments to try to explore this. And I would love to learn about people that study this and see what kind of reports they have about conscious experience while on drugs. I don't know. I know that in dreams, this does not happen. Are they sort of dreaming while I am on drugs? I don't know. Very, very good question. Thank you. I love it.
We should do EEG while people are on drugs.
May I jump in with a question to Pedro?
Yes.
Well, two observations. First of all, the default mode network is called default for a reason, in my opinion. Second one, there is the notion of perception as controlled hallucination.
Yeah.
So drawing these two things together, you could argue that control is not so much necessary for switching off the perception and going into a kind of internal mode. Perhaps control is just needed, or better, the spontaneous internal mode is perhaps just the default, and you need the control for operating in a more perceptual mode. That would imply something for consciousness, namely and predominantly that consciousness is always there in the internal mode, and that the perceptual mode is something that happens when the default mode gains something extra, that is, gains some control or the system gains some control over the perceptual systems. That means perception is far more active and exploratory, predictive perhaps, and therefore there is not necessarily the intervention of a higher order system in switching between the two. It's just devoting your attention and devoting your consciousness to the external world. What do you think of that?
Right. I think that it's not that you have consciousness like an item that you devote to one thing or another. Is the subjective experience to it's a higher level of it's not the qualia in general, but it's
no, I'm not saying that. Huh?
Right. Okay. But I agree with you in that consciousness may have different intensity depending of the coupling or uncoupling with the external environment. I don't really like this idea of control hallucination, what Anil Seth has put forward, because I think that we never have hallucination. We experience the model that our brain makes about the world. Our brain cannot really experience the world as it is. It's always a model of what happened that our brain builds with. Yeah. And this is not hallucination. This is what happened. Hallucination refers to something which is sort of false, and it's never false. It's always true. But anyway, but I will agree with you that in the default mode network, we may be our consciousness will be engaged in this mode, but will also relate to the fact that we experience this sense that we are doing a simulation and is different from engaging in the world. So we need to be aware, or consciousness will be the susceptible experience of an understanding somehow that we are in this mode and not in an engaged mode, in a perception mode.
Mm-hmm. I sort of agree with that, but I mean, it does mean that the control aspects are sort of deployed predominantly and mostly in the perceptual mode rather than in the default mode.
But wouldn't you agree that this control of this, where we have this consciousness, should be always present, whether we are in a perception mode or in default mode? Somehow the brain needs to understand where it is, because otherwise cannot control the switching between one mode and another.
Oh, certainly, but I think there is perhaps more control necessary in the external mode.
Maybe, but I would not. I understand your point, but I wouldn't know how to formalize this. Maybe you can suggest something on that.
Okay, thank you very much.
Thank you.
Well, we have reached the time scheduled for the end. I don't want to start a fight asking the global workspace or the predictive coding, whatever. So, but I will keep it like it is. I really want to thank each of you who came and presented to us your studies and reflections. Now I have many things in my mind. Thank you for being here. Thank you for the listeners who came from every part of the world. So thank you again, and please wait for the email that we will send to you, possibly with a link where we will publish the video of this, the registration of this event, and see you soon, I hope, with another event or something similar. I will contact each of you by email anyway.
And
Thank you very much.
Thank you a lot. Have a good lunch or dinner or whatever you are going to have now.
Thank you very much.
Thank you
for
very
the
much. It
invitation.
was very
Thank you,
Great to see
Kiara.
you all.
Bye-bye.
Thank you a lot.
Bye.
Bye-bye.
Bye,
Bye.
Andres.
Bye-bye.